Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. First of all, I would like to welcome you all to this great event. My Honorable Dr. Martin Wilkes, as invited speakers, the committee and all participants. Ladies and gentlemen, so now we have the session for presentation of our invited speaker, Dr. Martin Wilkes. Before that, let me introduce our invited speaker, Dr. Martin. Okay, Dr. Martin is already here, yeah?
Hello.
Yes, Dr. Martin. Good afternoon, Dr. Martin. Yeah.
Good morning from the United Kingdom.
Okay, yes, good morning. Okay, I will introduce Dr. Martin Wilkes. Dr. Martin Wilkes is assistant professor in Coventry University. He completed a master degree in University of Birmingham School of Geography, Earth and Environmental Sciences in River Environmental Management. And also he completed PhD in Environmental Informatics at University of Worcester Institute for Science and the Environment. And Dr. Martin is assistant professor in Coventry University from 2017 until now. And he also had some research, maybe it's too much research, yeah, about biodiversity, maybe, and aquaculture also. Okay, so now we will let Dr. Martin Wilkes to present his research. Please welcome Dr. Martin. If time is yours.
Thank you very much. It's a pleasure.
I will start the.
It's okay. I will share the presentation.
Okay, I will stop. It's okay.
Okay, let's see. Yes, it's a pleasure to be back at the conference for the second time.
Yes.
Thank you to Professor Mukhlisin.
Yeah.
Melissa, can you see my screen?
Yes, I can see.
Okay. And just to prove I was at the last conference, here is the, can you see the bag? Not really. Yes, you can see the bag, which
yes.
I still use to go shopping every week. So thank you for the bag.
Okay.
And thank you to Professor Mukhlisin for the invite today.
Yeah.
So if you can confirm, Melissa, that you can see my presentation, is that right?
Yes.
Wonderful. So my presentation today is titled Reading the Curve of Biodiversity Change. So we have many excellent looking presentations in this session about the very fine details of biodiversity. So this presentation is really about the general picture of biodiversity change, and I'm focusing on freshwater ecosystems. So why have I chosen the title Reading the Curve of Biodiversity Change? It's inspired by a paper last year from David Tickner and colleagues from the World Wildlife Fund in the UK called Bending the Curve of Freshwater Biodiversity Loss. And in that paper, they argued that the current trajectory of freshwater biodiversity globally was very negative. And we needed to implement a 6-point emergency recovery plan in order to bend the curve of freshwater biodiversity loss. But when we look more closely at finer scales, regional scales, we find that the trajectory of biodiversity change in freshwater ecosystems is often positive and more likely stable over longer periods. So hence my title, Reading the Curve of Biodiversity Change, pointing to the need to understand what is the current trajectory. And within that, there are some questions, some of which I hope to provide some answers to. First of all, can we identify which regions have declining, stable, or increasing species populations? Can we identify good and bad examples of biodiversity change and learn from them? Which species characteristics or traits make populations more likely to decline over time, and which traits make populations more likely to respond positively? So this is really what my presentation is about today. And it's in 4 parts. So in outline, the first part will be a discussion on global and clustered biodiversity trends, and I will explain what I mean by clustered biodiversity trends. Secondly, we'll think about the data availability and modeling techniques to assess biodiversity trends at large scales. And we'll apply those in a case study to rivers in England in the period 2002 to 2019, some research that I am currently finishing now. And then finally, we'll think about future prospects for global biodiversity monitoring. So as biodiversity scientists of one kind or another, I'm sure you will have all seen this graphic from the Living Planet Index, showing that there's been a mean decline of 68% in vertebrate populations worldwide. And when we look more closely at the continental trends, we see that not every continent has the same trend. We can see here that that trend is different depending on where in the world we are. And when we look even more closely, such as this paper from Leung et al., who looked across a wide range of freshwater taxa across the world. And actually what those authors found was that the Living Planet Index was driven by less than 3% of extremely declining vertebrate populations. And when these populations are excluded, the global trend switches to an increase. So that's a very different picture to what we get from the overall Living Planet Index. So this is an example of what I call geographical clustering, clustered biodiversity trends. But we also get phylogenetic clustering, and here's an example on amphibians from a paper by Novakovsky et al. In 2018, where we see that certain parts of the phylogenetic tree here, there are certain families with high numbers of heavily impacted species. That's the colors, the green and the yellow colors. So we see Hylidae, Buffonidae, and Dicroglossidae are particularly impacted by habitat change. So that's phylogenetic clustering, but we also find functional clustering. And here's an example on forest bird species from Newbold et al. In 2013, who looked across a gradient of land use change at different bird traits. And we can see here that the responses along the gradient are different in each trait category. So diet, migratory, and habitat generalism. So that's an example of functional clustering. So just to summarize on that part of the presentation, biodiversity declines are strongly clustered geographically, phylogenetically, and functionally. So my question is, how do we best identify those clusters most in need of emergency intervention? So the areas, the taxonomic groups, and the trait groups which are most in need of intervention. And of course, to do that, we need data. And this is what my group likes to do, is to work with lots of data. And we are very lucky as ecologists to be living at a time when more and more ecological data are available for analysis. So traditionally, a paper in ecology might have had around 100 samples, and maybe meta-analyses would have thousands of samples. My group works with data from the Environment Agency in England, which has hundreds of thousands of samples. And now we're seeing DNA-based technologies that can deliver even greater volumes of data. And then we have global repositories such as the Global Biodiversity Information Facility, GBIF, which now has well over 1 billion records of species occurrences. So we're very lucky as data-focused ecologists in my group to be living at this time when we have access to such amazing data. So some people think that this means that ecology is now in the world of big data. And I've put a question mark here because in some ways, biodiversity data are big data. In other ways, they're still relatively small. So we'll think about big data now. And one framework for thinking about big data is called the 4 V's. So they are volume, the amount of data that we have; velocity, how quickly data get from the field to your computer for analysis; and variety, so this is data from many different sources, collected using different methods, etc. And then finally, veracity. This refers to data with different levels of uncertainty. So when we think about biodiversity data, on the one hand, we have low volume. So this is typically gigabytes rather than terabytes or picobytes of data, and low velocity. So the data typically take months, even years, to be processed and reach us for analysis. On the other hand, in ecology, we're dealing with data with very high variety. So we might be using data from different studies that have used methods, different methods and different sampling strategies. And we're also dealing with very low veracity, so high rates of uncertainty due to sample bias, measurement error, etc. So let's focus on these two areas and think about how do we overcome these challenges. So in terms of high variety, we can use highly standardized monitoring data sets. And this is what my group has been doing using the Environment Agency river monitoring data in England, where I am based. And then the low veracity challenge, this is where we have potentially high rates of sample bias and measurement error. We need to use modeling techniques which account for that uncertainty. So let's move on to think about those modeling techniques now. And we start with a description of a basic species distribution model. So essentially, a species distribution model is a regression where we have some environmental data across the landscape and so a sample of a species distribution. And we put these things together in a regression model, and we have a prediction of the species distribution. Now, when we're thinking about biodiversity trends over time, we can also include time as a variable in these models. There are two issues with this basic approach, which we can use on the one hand to predict population distributions, so the presence or absence of a species across the landscape. We could also use them to predict population abundance, so the total population abundance of a species. And there are problems with each of these approaches. So when we're modeling population distributions, we have to be mindful that we have not perfectly observed the distribution in space. There is always a spatial bias towards certain areas or certain environments. And on the other hand, when we're modeling population abundance, we have to remember that we're not perfectly detecting the number of each species present in the habitat. So I'll just explain a little more about how we overcome these challenges, starting with the challenge of population distributions. And to overcome this challenge, my group uses hierarchical modeling of species communities. So this is a hierarchical Bayesian species distribution model, which can actually be applied on multiple species simultaneously. So it's a kind of community level model. And the main way that this helps us overcome spatial bias is through controlling for the spatial and temporal context using metadata about the samples that we have. So this would include spatial structure, spatial coordinates, river basin, time and date, so on. And then the other data that we need for this model are our environmental predictors, and that may include time if we're interested in biodiversity trends. We need a phylogeny, and we need a trait database. So if any of these concepts are new to you, or you have any questions about them, please do ask in the question and answer session at the end. And in terms of population abundance, then, where we have this problem that we're not perfectly detecting the presence and the number of individuals, we use zero inflated models. So zero inflated models are really a two-part model where we model the number of zeros in our data separately to the actual count of individuals, and then we put those two models together. So the zero part of the model is there for when we go out to survey a habitat to record the number of individuals, but for some reason we missed this, in this case a hippo, we missed the hippo, it was hiding under the water. Or the other reason that we might get a zero here is because just happens to be that at the time that we went out to this site, the hippo was not there, but otherwise the hippo lives there and the habitat is good. And then in the other part of the model, we get the genuine zeros. We went to the site, we did not see a hippo, and indeed the hippo does not live there because the habitat is not good. And then finally, we have the part of the model where we predict the number of individuals, greater than zero, of course. Typically, these models are fitted using traditional frequentist techniques, so a typical regression model. But if you're anything like me, you tend to see these diagnostic plots where we have deviation from normality, and so there's a lot of structure in the residuals. So what my group uses are these Bayesian models, and particularly the BRMS software package in R. Again, if you're interested in more details about any of this, please ask in the question and answer session. So let's apply those ideas now to a case study in my region, England, over the last 20 years. So the background for this is that a paper back in 2012 found that invertebrate communities in freshwater in the region had increased or improved in terms of species richness between the 1990s and the late 2000s. And then a subsequent paper last year found that the types of species which had increased the most in the region were the kinds of species which are very sensitive to water quality or poor water quality. So the conclusion so far, just looking at invertebrates, is that there's been an improvement in biodiversity because of work that the government and charities have done to improve water quality in rivers. So that's a happy story from England about biodiversity. So we wanted to look across all four major taxonomic groups of rivers using data from England. So we looked at macrophytes, single-celled algae or diatoms, invertebrates, and fish. And if you look at the number of records that we have each year, you can see that this is a lot of data. So I think in total we have over 200,000 observations of community structure in rivers. And this is quite a small country, I'm sure you will agree, particularly if you're from Indonesia. So let's have a look at the results that we saw in terms of increases in species richness or decreases in species richness across the country. So here for each group, we have the probability that the taxon richness in that group increased or decreased. But in every case here, we can see there's a high probability that the taxon richness or species richness increased in the period 2002 to 2019. What we can see in the maps next to each panel here, that there are some river basins where species richness declined. So here's an example of identifying the geographical clusters which are most in need of intervention. Let's look at phylogenetic clusters now, and here are just two examples. Macrophytes on the left and fish on the right. So in red are species and river basins where we see a decline. In blue are increases in the distribution of each species. And we can see that there are a number of species which are both declining and increasing. And on the right-hand side here, we have the phylogenetic tree. Now, it's important to note that in our results, we did not find any significant phylogenetic clustering in the data. So this suggests that there are some other processes going on. And we think those processes are due to species traits. So let's look at some functional clusters now. I won't describe in detail what these trait results are telling us, but what we see are declines in the following traits groups. So we see declines in species with high resistance to floods and droughts. And we also see the loss of species characteristic of higher nutrient concentrations. So this again supports the idea that there has been improvements in water quality in the region. If we look at invertebrate and fish data now, we can see that there were declines in larger bodied species. So smaller bodied species did better over the period. We saw losses of species with a range of resistance strategies. And we also saw declines in predators and parasites. So in summary, there is a high probability of both richness and total abundance increases. I didn't show you the results for total abundance, but that also increased on average across the region. However, some river basins did show decline. So that's an example of geographical clustering at the regional scale. But there was no significant phylogenetic clustering. However, there was strong functional clustering. So we saw changes in the expression of traits based on resistance to disturbance, resilience, and trophic status. And these patterns suggest widespread improvements in water quality, but changing disturbance regimes. And we think that's due to extreme events. We see more and more heat waves, droughts, and also extreme floods in England. So I'll quickly move on now. I realize I'm running out of time. So the final part of my talk is to think about future prospects for global biodiversity monitoring. So here's a graphic from the Global Biodiversity Information Facility online, the number of species presence records that are available across the globe. So the deeper red colors are where more data are available. And you can see that there's a global bias here, which isn't typical and not really surprising to us. The other problem with this data is that we have very limited ability to infer trends over time from this type of data. There are other databases which are growing, such as BioTime, which include species presence, absence, and abundance data. But again, from these maps, you can see that there's a bias in the global distribution of the data. But I do have good hopes for global biodiversity monitoring because the data resources are already gathering to fit these kinds of models. And we're just showing some examples of the sources where we can go and get these data to drive the models that we're using on almost a global basis, albeit with spatial bias. And we also have a fantastic group in the GeoBond group. So this is the group on Earth Observations Biodiversity Observation Network, who have been thinking very, very clearly about the needs for infrastructure to support global biodiversity monitoring. And if you're not familiar with their work, I would recommend having a look on their website. So to achieve a situation where we have equitable and even distribution of global biodiversity monitoring, we need to firstly expand coordinated monitoring efforts through GeoBon and also keep contributing to repositories such as GBIF and Biotime. We need to harness DNA-based monitoring and citizen science, but not as a replacement for professional manual sampling. And here are some logos from some organizations which are leading the way in that regard. And third and finally, we need to train a new generation of biodiversity scientists in the key skills around data management, statistics, and programming. And I'm showing my email address and WhatsApp number here in case anybody is interested in talking to me about these aspects of training. And if I can ever be of any assistance to you and your graduate students, it'd be a pleasure to hear from you. So I'll hand back over now to Melissa for question and answers.
Okay, thank you, Dr. Martin Wilkes. It's very informative and interesting talk. Maybe any questions from our participants? You can ask directly by click raise hand or type in chat room. Yeah. So Dr. Martin, it was interesting that you also provide the train about for the graduate student also, yeah.
Yeah, I'm always happy to help and provide training and support for any students that want to develop their ability to look at big data sets in biodiversity. But I realized that, doing this kind of work in England, where we have such a large amount of data, it's different concept to perhaps in Indonesia, where at least in fresh water, you have data, but not the same volume of data that we have here. So this is why I wanted to talk about future prospects for global biodiversity monitoring, because when we have an even distribution of monitoring globally, and this is when we can really begin to understand how we affect biodiversity on a global scale.
Yeah, very interesting. Okay, maybe Mr. Zulkarnain.
Okay.
Yeah.
Thank you.
Yes.
Assalamualaikum, Dr. Martin.
Waalaikumsalam.
MashaAllah. Alhamdulillah. Very happy to meet you again.
Alhamdulillah.
Alhamdulillah. So, okay, saya mau cerita sedikit nih tentang Dr. Martin. Juni 2019 beliau menjadi pemateri di Icfaes yang pertama di Fkp, dan beliau hadir langsung ke Banda Aceh waktu itu. Nah, setelah selesai konferen, beliau kita bawa ke sana kemari untuk melihat beberapa kawasan spiritual di sini, dan beliau sangat tertarik sekali. Selepas itu beliau kembali ke negerinya, ke Inggris, dan Februari 2021 yang lalu beliau mengirimkan WA ke kami dan beliau menyampaikan sudah mengucapkan 2 kalimat syahadah. Jadi ini yang ingin saya sampaikan kepada kita semua, bahwa dengan keberkahan daripada negeri kita ini, maka Allah SWT telah berhidayah kepada Dr. Martin untuk mengucapkan 2 kalimat syahadah. Ini saya ceritakan karena tadi saya menyampaikan Assalamualaikum kepada beliau. So I just told the history about the last time, the first Icfaes, which you as the keynote speaker in this conference, and then you compared to Islam. Alhamdulillah, may Allah bless you and your family as well. So actually, in this event, I have no question actually, but I just wanted to how to say that I'm happy to meet you, and then may Allah bless you with your family. Happy to meet you in this forum, even in virtual forum. But I hope that I can visit your country one day, inshallah. Okay.
Inshallah.
That is all from you. Nice to meet you. Thank you.
Thank you,
Assalamualaikum.
Paksul. Assalamualaikum.
Waalaikumsalam. Okay.
Maybe it's like our union. Yeah, Dr. Zulkarnain.
Yes.
Okay.
Thank you.
Bless you,
Any-
Paksul.
Amin.
Anybody else?
And I have
Yeah.
question.
Yes. Yeah. Belita Torina. Yeah.
Is it
I
okay?
think the other
Yeah,
guy
it's
has
okay.
another question. It's
Oh.
first than me.
Thank you very much.
Okay, okay.
Dr.
I
Martin.
will go on after him.
Okay. Okay, please. Who is ask the
Professor Kasim.
Okay, Professor
Okay,
Kasim.
you have a question?
Yeah.
Because yeah. Okay, thank you very much,
Yes.
Dr. Martin. As your presentation is very nice presentation that for the biodiversity. But mostly your information related to biodiversity is in England. That is, I mean, that is cold one. I mean, the season is cold, and that is really different with the tropical one. Do you have any idea related the tropical one? I mean, that is the trend of the biodiversity, particularly in tropical forest or in tropical area. Compared to subtropic or in temperate ones, the trend that you give information.
Yeah, so I don't personally have any data or record about trends in tropical systems, but looking at the literature, you can see that there are examples of both negative trends and positive trends and many apparently stable systems in terms of biodiversity. I'd encourage you to look at the paper by Leung et al. And I'll just tell you now which journal it's published in. So that's published in Nature, and the title of the paper is Clustered versus Catastrophic Vertebrate Declines. So they looked across tropical, subtropical, temperate, Arctic systems, and they found examples of increasing biodiversity in all of those systems. But of course, there were some populations which are experiencing catastrophic declines. So whilst I support the idea that global biodiversity is declining, what I wanted to do with my presentation is show that biodiversity in freshwater systems is not declining everywhere. And if we're going to address the problem of biodiversity loss, we need to be good at identifying the places and the taxonomic groups that need our emergency intervention the most. So what I would love to do in the future is apply the kind of analysis I did in England, but to the whole world. The problem is, of course, the variety of data and the spatial distribution of data is currently, we don't have a good enough distribution of data to be able to do that without further assumptions. But we can look to excellent papers such as the paper by Brian Leung et al. Published in Nature last year, which I encourage you to go to that paper because there is information about tropical systems there.
Thank you very much.
I will share the link on the chat.
Okay, I'm waiting.
Was there another question?
I think it's from me.
Yes.
Okay, thank you so much. But before correct me if I wrong or I misunderstand about it, because at the beginning you said at your presentation you said that there is decreasing in the biodiversity of larger invertebrate. Is it true? And then
Yes. So in our case, in the data set from England, so this is a temperate system, as mentioned by Dr. Karim. We found that that was a very strong pattern, that larger bodied invertebrates and fish were, I wouldn't say decreasing, but I would say the trend was more favorable to the smaller bodied species. So what we think is this is a general pattern of increasing resilience in ecological communities. So if an animal has a smaller body, it typically has a higher fecundity and a faster reproduction rate, so is more resilient to disturbances. So if there is a disturbance in the system, that species can bounce back, recover more quickly than the larger bodied, less fecund species with the the longer reproduction time. So that's the pattern that we found. But I realized that in tropical and subtropical systems that we might find something very different in terms of body size responses to climate change, for example.
Yeah. So
do you have any information about that?
No. I mean, I agree with you, but then I'm a little bit confused whether it's a different species. I mean, the large species kind of, let's say, decreasing, and then the small species, let's say, survive. But then, is there any information about these large species become a smaller body, for example? Has a smaller body because they kind of adapt to the situation, for example, the predator or whatsoever?
It's a very good question, because what we did in this study is we looked at trait information from databases. So we just took the species name and then we took the range of the traits, for example, body size from a database. But what we can do for many of the data that we have is to look at the actual measured body size of the fish caught in each site. So that's what I would like to do in the future, is to look at the actual body size data over time to see if statistically we can find a difference in the body sizes within a species. So it's a very good question and something that we would like to work on in the future. If you'd like to work on it with us, you're very welcome to become involved.
Thank you so much, because I also interested in the microplasticity, and then also want to see whether there is a microevolution in this organism size. So it will be interesting if we can work together. And then I also have, I'm so happy when you say that you are very welcome if somebody contacting you regarding the big data and how to handle them, especially to work with R. Because now I kind of working with R, and to be honest, R is killing me. It's so difficult.
I've been there, yeah, but I have a long relationship with R, and with time it gets easier and you become more confident and you become the master of R instead of R being the master of you. So stay with it, and please get in touch with me and we can exchange some best practice in R programming. Yeah.
Yes, so lovely, because in these last 3 years I work with R, and still kind of the situation is the R mastering me, not I'm mastering the R, as you said. So thank you so much. I will contact you.
Yeah,
Would
thank
be a pleasure.
you to Martin. Yeah, it was interesting for Mbak Pelita, yeah. Maybe you can join with Dr. Martin, yeah, in the next project. Sorry. And the next question, maybe the last one from Mr. Dedi. The biodiversity in the whole world is going to decreasing. Many effort has been done to overcome the problem. What is the simple and cheaper solution to solve this problem, especially for small or poor countries? Yeah, Dr. Martin, maybe you can answer these questions. Maybe you are.
Thank you, Dedi, for extremely difficult question to answer. Of course, of course, there are no easy or cheap solutions to the problem, because the problem has so many different faces. So we have, on the one hand, a large-scale global change such as climate change. On the other hand, we have very local problems with specific pollutants and everything in between. So, I know in the region around Aceh, there is a problem with marine and freshwater plastic pollution, for example. And that's just one example of a problem that impacts biodiversity. So there's no easy, cheap, and fast responses to this. But I think the emergency recovery plan that was mentioned in that paper from David Tickner and colleagues has some very good ideas. And I think that forms really the basis of the solution that you mentioned. It's not easy and it's not cheap. But again, I will post the link in the chat if you'd like to follow up there. But really what I wanted to talk about in my presentation today was actually one of the first steps that we need to take to understand how to help biodiversity globally is to understand where it is declining most, which taxonomic groups and which species trait groups are the ones which most need intervention. So that was just my small part of the solution, but I encourage you to go to that paper by Tickner and colleagues, which is in Bioscience, and I've just posted the link in the chat. Thanks for the question. I'm sorry I can't answer very well.
Okay. Yeah, thank you, Dr. Martin. It's very interesting talk. Yeah, it's about the big issue in biodiversity, yeah, globally. Okay, maybe at the end, before we leave this session, maybe we can take picture first. Yeah. Okay. 1, 2, 3. Yeah. Wait a minute. 1, 2,
Cheers.
3. Yeah. Okay, thank you very much, Dr. Martin Wilkes. So now we will move to the oral presentation session. And Dr. Martin, if you have time, you can join with us, or if you have another work, it's okay. Yeah.
I would love to stay and listen to the presentations. Yeah.
Yeah, it's very interesting. Okay, maybe we will start for the first presenter. I will call Ali Fikri Ramli. Yeah, it's already here. Yeah, I will share the PPT, or you want to share? Yeah, Mr. Ali. Okay, maybe I will share the presentation. Okay, wait a minute. Yeah, yeah, Mr. Ali, you already shared? Oh yeah, it's okay. Okay, please welcome Mr. Ali Fikri Ramli. Oh yeah, it's only 7 minutes for presentation and 3 minutes for questions. Yeah, okay, thank you.
Assalamu Okay. Hello, Assalamualaikum and good evening to all the committees,
Waalaikumsalam warahmatullahi
moderators, and
wabarakatuh.
also all the presenters. So I'm Ali Fikri Ramli, and today I'm going to share to you about my review on the enhancement of nutritional value on zooplankton by alteration of algae media composition. So the introduction for this review was, as we all know, aquaculture, sometimes referred to aqua farm, is among the fastest growing food sector in the world. And the effectiveness of aquaculture farming was critically determined by the nutrition and feeding, which must be improved to the sustain of aquaculture. So the critical was dominated by the feeds and fertilizer source. However, to finding the acceptable larvae feed has been significantly challenged, and most feed do not match nutritional requirement for the larvae to consume. And as a result, live feed, particularly microalgae and zooplankton, have been chosen to be suitable for the larvae growth in aquaculture sector. Other than that, zooplankton is essential natural food source for the diverse range of aquaculture species. For example, fish species, mollusk larvae, and crustacean larvae, which is derives the nutritional predominantly on zooplankton. So the purpose of this review is to gain a better understanding on how increasing the nutritional value of zooplankton by enriching with microalgae with alteration nutrient composition, and how the improving micronutrient can affect the nutritional value as required by the larvae. Previous research from Kandati mentioned that the enrichment technique is carried out in hatchery in order to provide more nutritious live feed with a big scale in terms of protein and other nutrient content. After that, from research from FAST also mentioned that microalgae, fish meal, and cannoli are most often being used for the enrichment, which can increase the HUFA and also important amino acid content. We go to the enrichment of the plankton with microalgae. So basically, microalgae feeding is an effective method of increasing the nutritional content of zooplankton. Previous research from library mentioned that microalgae fed with profile-rich diet was successfully increased the nutritional content in zooplankton due to the fact that Moina sp and copepod are filter feeders, so they graze on the phytoplankton bacteria or the tritoph, and this plankton primarily enriched by can be enriched by microalgae species. Research from Rusty mentioned that Moina and copepod enrichment are not fully being used in aquaculture. Even though this zooplankton has rich protein content in its nutritional composition, however, enrichment via microalgae can be used to ensure that species mass culture is sustainable and could be used for the larvae development, especially and also for the other rotifer and copepod culture. Other than microalgae, the use of lipid emulsion also been used in enrichment to increase the nutritional content. Highly unsaturated fatty acid has been successfully in large growth of Moina macrocopa. According to the study from Singh, Microbrachium bosenbergi, or giant freshwater prawn, the larvae was grown and survived increase when the feed with Moina enriched with Mufa obtained from microalgae. As you can see in table 1, this is the list of the current microalgae species have been used as enrichment in aquaculture study. For the first study from Azani and Rusdi mentioned that chlorella has been used to enrich Moina microra and Cyclopod copepod for the growth and coloration of ornamental fish, which is Siamese fighting fish, or in Indonesia we call it cupang, and also angelfish. Then Nanochloropsis salina have been used in the study of Danish, which is enriched to enrich the mix of Branchinus plicatilis, which is rotifer, and also Artemia and been tested on the growth, carotenoid, and pupa of clownfish. Other than that, Tetraselmis susica and the meat of Cetosuros cartifran has been used to the enrich of Artemia napli for the development or survival of river prawn from Mendes.
Sorry, Mr. Ali, it's 2 minutes left.
Okay. And we go through the nutrient content of microalgae. So the nutritional content of microalgae species will vary subsequently. Microalgae diet potentially to improve the nutritional value of zooplankton in the numerous factors, including nutritional structure, form, digestion, and biochemistry content. Because microalgae are principal production in aquatic food, and we can replicate that in the aquaculture industry, which they can offer EPA, AA, and DHA, which is required to grow and development of aquatic organisms in aquaculture. For example, carotenoid being used in pellet. Microalgae can supply this necessary pigmentation to the color for the several ornamental fish. The key ingredient content in microalgae can give zooplankton and fish larvae, for example, amino acid. Amino acid composition of microalgae generally identical and can affect the growth circumstance. Microalgae nutritional content also can be altered when N will represent the stress, for example, nitrogen and phosphorus limitation, which can sustain maximum growth in aquatic organisms. So the relationship between microalgae media composition to the nutritional value of microalgae growth. The culture medium chemical composition was complex. The more complex the fatty acid profile in the microalgae, and the more nutritional zooplankton or fish larvae can be due to direct or indirect intake. Then Rusty also mentioned that nitrogen and phosphate can manipulate the ratio of this nutrient in the percentage of polysaturated and fatty acid purifier. And additionally, culture medium can be improved in terms of nutritional value as a feed for zooplankton or microalgae. So the number of studies has demonstrated that fertilizer availability can
Sorry, Mr. Ali.
Okay.
The time is over.
Okay.
Yeah. Maybe the next, or maybe you want to explain about the conclusion? Yeah.
Okay.
Yeah.
So the conclusion for this review was the outrage influence on zooplankton quality by altering the media composition of the microalgae, which is this micronutrient are minor factor that increase the quality of microalgae and also can increase the quality of zooplankton. Other than that, critical nutrient of water also can be a major fertilizer supply to the musculature of microalgae. And lastly, microalgae enrichment serves to increase the nutrient content of live feeds, such as fatty acid or other compositions. Thus, by entering and enriching microalgae mediated phytoplankton can have a positive impact, especially larvae breeding and also aquaculture development. That's all. Thank you.
Okay, thank you, Mr. Ali Fikri. Maybe we will move to the next presenter. And for the question, you can ask after the third presenter. So each three presenter, you can ask the questions. Yeah. Okay, the next presenter, Miss Amira Yuslan. Yeah, please welcome Miss Amira Yuslan.
I will share the screen.
Yeah, I will share.
I will share the screen.
Oh, okay. Wait a minute. I will stop. Yeah. Yeah, you can share the screen, Miss Amira.
I had shared the screen.
Yeah. It's only 7 or 8 minutes. Yeah.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum and a very good evening to all of you. My name is Amira. Today I would like to present my research on the title, The Effects of Enriched Cyclopod Copypod on the Coloration and Feeding Rates of Beta Splendors. As the introduction, the beta splendors are often known as yummy fighting fish or beta fish, and the productions of beta fish are depends on the nutrient that we give during the feeding. The body colorations of beta fish are highly dependent of enrichment zooplankton, which is used as a food. The diet provided in high nutrition can improve the age reproduction and pigmentations of beta fish. The pigmentation, such as carotenoid, are important for coloration and also to increase the beta fish industry. The live feed that we use as food for ornamental fish, such as copypod, cannot itself synthesize the carotenoid pigment, but we can get from the enriched copypod with potential diets such as microalgae. Okay. Therefore, my study was conducted to determine the influence of feeding enriched copypod on the color, growth, feeding rates of beta splendors. Okay. For the material and method, this experiment was conducted at University Malaysia Terengganu Hatchery. The copypod were sampled at situ waters Terengganu, Malaysia, and the copypod were cultured at optimum parameter, which is temperature at 26 degrees Celsius, salinity 10 to 50 ppt. Okay. For the treatment diet, the diet that I have used is chlorella, capsicum, mixed vegetable, which is carrot and spinach, yeast, and rice bran. All the feeding diet will be fed daily to copypod culture, and the culture will be fed 2 weeks with a specific diet, which is T1 until T5, before I conducted this experiment. The concentrations of the diet that I use is 5 milligrams per liter. Okay, the proximate analysis of copypod, of enriched copypod, were conducted also at UMT hatcheries. All the samples were analyzed in 3 replicates by using standard protocol by using from AOAC. The sample of enriched copypod were harvested and sieved by using plankton net, and the group protein were analyzed by using Jadal method. For the feeding rate used is 10 to 1, which is 10 copypod per individual to 1 one fish. And for the coloration were assigned in the bodies of 5 randomly selected fish by using conical monochrome chroma. For the data analysis, the data were analyzed by using one-way ANOVA with the key test. Okay, in short, this is my experimental design view, which is copy pot will be cultured and maintained. Then the diet will prepare. Then the nutrient analysis, which is lipid and proteins, were conducted. Then the enriched copy pot will be fed to beta fish, and the parameter that observed is coloration, feeding rate, and growth performance of beta splendors that fed with enriched copy pot. Okay. For the result and discussion, the proximate analysis of enriched copy pot, which is cyclopod copy pot. The protein and lipid content of copy pot were significantly impacted by different dietary arrangements that they used. The highest protein content were recorded in treatment 1, which is copy pot enriched with chlorella species compared to other dietary treatment. And the higher lipid content were recorded in treatment 5, which is copy pot enriched with rice bran. These results were comparable with the real study, which is the protein content in copy pot, which is Acacia species, Centropes species, and Erythromora species are between 38 and 56%. And the lipid content is between 6% to 22%. The highest in protein and lipid contents of enriched copy pot can enhance the beta splendors growth performance and survivability. Okay. For the growth rate performance of beta splendors, the growth rate and survival are impacted by difference of diet given. The highest specific growth rates and survival were recorded in treatment 1 compared to other dietary treatments. This result can be spotted by previous study, which is a recent study found that juvenile beta splendors that fed on live feed, which is Moina micrura, supplemented with chlorella, result in high growth performance and also survival rate.
Ms. Amira, 2 minutes left.
Okay. For feeding rate, the optimum feeding rate were directly proportional with frequencies of the diet. This means the sufficient feed frequency can lead to highest feeding rate, which made it easier for ornamental fish to manage and market it. Yeah. As the conclusion, the effects of enriched copy pot on the coloration and feeding rates of beta splendors was successful as the arrangement type given, which means all the parameters were impacted by different diet arrangement. The copy pot enriched with rice bran produced high protein content, and is comparable with result of copy pot enriched with chlorella. This shows that the use of agro-industrial residues such as rice bran also can be substituted microalgae culture and supplemented together with microalgae, which can boost the productions of live feed and also fish larvae culture. Seeing the synthetic carotenoids are quite highest in price and also have limitations, cheap and readily accessible sources such as rice bran, carrot, spinach, and the arrangement method to copy pot before feeding to beta splendor arrangement. The findings reported in the study may serve as guideline for further investigation. The developments of enriched medium for copy pot to improve the growth performance, fiber, and also the nutritional status of culture organism. That's all. Thank you.
Yeah, thank you, Ms. Amira Islam, for the interesting topic. Okay, next we will move to the third presenter, Mr. Ma'ruf Kasim. Yeah,
Thank
please
you.
welcome Mr. Marcus.
Okay.
Yeah.
Thank you very much, moderator, Bu Siska. Thank you very much. I cannot be changed my background because while I change it, the background was cover my face.
Yeah, it's
So,
okay.
okay, I'm
It's okay, never
just
mind.
present. Okay, can I share right now?
Yes.
Appears? Okay.
Yes. Yes.
Okay, thank you very much, ladies and gentlemen. It's very pleasure to have a presentation in this international symposium. I'm very glad to get invitations, even though I was submitting abstract. The title of our research is the existence of the epiphyte on the Thallus euchemadendiculatum. This is one of the seaweed in the faring depth, cultivated with the vertical net. This is one of our innovations patent. We have 12 patent in cultivations, and then this is one of our patent. We are from the Faculty of Fisheries and Marine Science, Halal University, Indonesia. It's far from Aceh, that is in southern Indonesia. Okay. For the background of our research is, as we know, that the seaweed cultivation is mostly likely cultivated in people coastal area. They cultivate the seaweed because they very simple method. They can cultivate the seaweed during 45 days, and then they can harvest it and then get the money. Just 45 days they can cultivate the seaweed. Then that is very simple method to cultivate it. Because of the very simple method, they are using the long line, using the tie, and then just use a bottle plastic, and then tie the seaweed, particular the Euchema denticulatum, and then let it until 45 days. But the problem is the epiphyte attached on the line, not only on the line, also on the thallus. So this is really affect the growth of the seaweed, because sometimes the abundance of the seaweed, the epiphyte, is much higher than the epiphyte. The seaweed. So that's why this is the most problem for the seaweed cultivations in particular in our side in Southeast Sulawesi. So we done this research to know the existence of the epiphyte on the thallus of the Euchema denticulatum particular. But aside that, this is the face of the problem. This is the problem really of the seaweed farmers in Eastern Indonesia. They have Icfaes disease. Look at this one. This is the epiphyte cover. That is cover the seaweed, so that's why this effect. And then another one is epiphone, and this is the method. Our method is we are using this research just in the coastal area located in Bau-Bau. One of the area we name it Lakeba. That is very good location for the seaweed cultivations. This is in the Southeast Sulawesi. This is one of Indonesian seaweed development center, because a lot of people cultivate the seaweed in this area. So that's why we done the research in this area. And this is the locations, as you know, that is in center of Indonesia, Sulawesi, and just 120 kilo from our university, Halal University. We have this is in Bau-Bau. This is because of this area is really nice for the seaweed cultivations. Very nice area, and then that's suitable for seaweed cultivations. This is our method. This is the Vertinet. This is one of our patent that we developed to cultivate the seaweed one. Why that we have developed this one? Because that is the grazing of the seaweed. It really occurs in every day. Not only like the medicine get from doctor 3 times a day, but every time if you cultivate the seaweed, then it will be grazed by the fish. Not only fish, but also sea turtle. Sea turtle is really like seaweed. So that's why to increase the possibility of the success of our research, then we have to using the Vertinet to protecting the seaweed from herbivore. And then we are try to collecting the 40 thallus meticulatum in 100 gram, and then place it in the floating cage, that is we call it a Vertinet in different type, and then we are try to not clean up, because we have to collecting the epiphyte. We have to collecting the epiphyte to to identify the species, and then to not counting the number, to weighing the weight. And yeah, that is every 10 days we have to calculate it. And the data analysis, we have to collecting the specific growth rate to analyze that, and then using the SPSS. Okay, we found that they have like more than 7 species of the epiphyte that the first what is we call it like occurs in all day and then all depth, that is Chromatophora crassa. That is that in the video in the picture before that we cover, I show you the cover of the seaweed from the epiphyte. That is one. That is like I mean that is like what is this? That is like they cover seaweed entire the seaweed. And then another one is the unidentified one, and then that is what the color is the white one, but it was occurs in all depth and then all treatment of our research. And we found that the Chromatophora also occurs in the depth 50, 100, and also 200. That is occurs in all depth of the abundance of the macroepiphyte. Chromatophora crassa, that is really, really much more compared than another. We found like Padina and then like Actopora, Sargassum, that is very few. But the Chromatophora is even in days one, we have a lot found that. And then another one, and in depth of in 20 days for another, that is Chromatophora occurs. So that's why this is really, really problem for the farmers. That is compared than another like Padina, Actopora, and Sargassum, that is really, really high. Almost 250 individual attaching on the seaweed. That way. And then the third day, that is occurs again. So all the day during our research, the Chromatophora crassa, this occurs compared than another. So this is really, really become the most problem for the seaweed that for the particular for Euchema denticulatum. That is the, we call it like attachment rate. That is we can count it. What is the rate of the attaching one? That is Chromatophora is really high. There's 150 in the first day, and then the last day that is almost up to 100 attaching on the Capapicus. I mean that the Euchema denticulatum, and that is we try to compare the environmental parameter, and then also with the another locations that we have found that in our locations is a similar rate compared than another one that is suitable, really suitable for seaweed cultivations area. So that's why the problem only is the epiphyte one. And we try to compare that is from the temperature, brightness, dissolved oxygen, nitrate, salinity, phosphate, and current velocity. We found that the epiphyte attachment, particularly the Chromatophora, that is they have significant with the current velocity and also the salinity. And that is the conclusion that macroepiphyte attaching on the thallus of the Euchema denticulatum, that is all species, but the one species that is occurs. The highest and lowest attachment rate in 50 centimeter and 200 centimeter is occurs in 356 and 223 individual per meter per day. That is the rate. And then epiphyte attachment of the Euchema denticulatum was more commonly found in lower depth. And then the based on correlations, measurement, temperature, and salinity was positive effect for the macrophyte attachment rate. Thank you very much for our presentation. This is our picture during our research, and thank you very much for Department Kementerian Riset dan Teknologi, and particular now Dikbud. Thank you very much, Bu Siska.
Thank you, Mr. Ma'ruf Kasim. Yeah, after 3 presenters, maybe is there any questions for the topic, for the sum topic today? Yeah, you can raise hand. Yeah, Miss Pelita Octorina.
Okay, thank you. I have 2 questions actually. The first one I delivered it to Fikri and Amira, I think they
Yeah.
work together on the same thing. So maybe this one question could be answered by the two of them. It's about they work with the enrichment of the algae, then give it to the zooplankton, and then they take a look of the effect to the fish, right? But then my question is whether it's information. I mean, this you two have any information about what is the effect of the nutrient enrichment to the zooplankton itself? Because then I'm really interested in zooplankton size, whether this nutrient enhancement change their morphology or their life history or something.
Yeah.
And then the second question I deliver to Mr. Hasim.
Maru.
Do I
Mr.
spell
Maru,
it correctly?
Yeah.
Hasim?
Maru.
Yes, exactly. I'm so
Maru
interested
Kasih.
with your talk. It's so passionate with so a lot of spirit. And then I also interest in your practical net.
Yeah.
It's really
practical net.
interesting because
Yeah,
I also work
practical net.
with plantation, and I want to, how to say it in English, to put I mean,
Yeah.
I work with the hydroponic.
Yeah.
So this is quite interesting in me because I never see something like it. So I could, maybe I will apply something like this. But then, yeah, I ask your permission first. Maybe I could use this, or probably it's your finding, right? So yeah, I let you know. I'm really interested in this. And then my another question is regarding the seaweed. I'm not really understand about seaweed because I'm not work with them. But then my question is, is depth give an effect to the biomass of the seaweed or the successful of seaweed cultivation? Because you have 3 depths, right?
Yeah.
And then I just interest which depth that give the best result. Thank you.
Okay, thank you, Ms. Belita, for the questions. Maybe for the first, Mr. Ali Fikri and Ms. Amira, maybe you can answer the first questions about the impact of nutrient enrichment for zooplankton. Yeah, this, yeah.
Yes,
Ms.
thank
Amira.
you for the question. Okay, since I did 2 copy part, I will explain in 4 copy part. Okay, in copy part, according to my knowledge, for the study, I found that unenriched copy part range of proximity analysis, which of specifically proteins is the highest risk of protein is like 50 to 60%. But when I enrich with the potential diet, it will reach to more than 70% of protein content. And for the morphology process, I will further conduct the experiment to analyze the further effects on enrichment to the copy part, to the zooplankton. So thank you.
Yeah. And maybe Mr. Ali Fikri, do you want to explain something? Yeah.
Thank you for the question. So basically, enrichment is being done to the zooplankton for their nutritional content before we feed to the larvae. So the enrichment is the term for the diet of zooplankton in the culture. Before we feed zooplankton to the larvae, the enrichment will be done before about 24 hours or earlier to make sure that zooplankton can absorb the nutritional content from microalgae before it can be transmitted to the larvae or fish or crustacean. That's all.
Yeah. How was the Miss Belita?
So you give the nutrient before the zooplankton given to the fish
Yes.
on a short time.
Yes, fine.
So you don't have time to analyze what happened to the morphology, right?
No.
Okay, I understand. Okay, thank you for the information.
Yeah, thank you, Mr. Alifuke and Miss Amira. And next, maybe Mr. Ma'ruf, do you want to answer the question for Miss Pelita?
Okay, thank you very much. It's really very nice questions, Ibu Pelita. Thank you very much for the questions. That I'm very glad to get these questions. That that is the vertical net. We call it one of our. We have 12 patent for seaweed cultivations. One of the patent is the vertical net. Vertical net we call it Vertinet. We are using the water column in the depth, so that's why sometimes mostly the people using long line one. If you're looking at the seaweed cultivations, they are just using the line one and using the used bottle plastic. So that's why a lot of garbage, a lot of floating in the ocean. So we are using the tape, very simple method. That is, we call it Vertinet, using like the picture like I saw you before. That is the 4, and then we are just using in the depth. So the depth is maximum in 400 meters, but in this reset we are using only 200 meter. So I will answer your question that the first up to 100 meter, that is the very good for seaweed cultivations, because irradiance, that is the decreasing of irradiance is affect with the growth of the seaweed. So if the seaweed can get the to activity, photosynthetic activity, then the increasing of the depth of the increasing of the growth of the seaweed, that is significant compared than another depth. So in 50 and 100, that is the good depth for the seaweed cultivation. So I allow you to using our patent. So just using it, because that is very good for avoid the herbivore. I mean, you know that is, if you use for seaweed cultivations, that sometimes herbivore like sea gannet or sea turtle is remain in the line. They graze all the seaweed, so you only get the line. So that's why if you are using the what will we call it, vertical net or horizontal net, if you need, I will put my email, and then I will send you all our patent, 12 patent, then we can use that. This is for who want to develop the seaweed cultivations, or for the research. That is we are pleasure for to our patent for using it. So that's why that is the seaweed. That the seaweed is mostly cultivated by people that because they are containing carrageenan for export one, for the raw material for all the industry, even for the food, for the cosmetic, even for the medicine. That is for the raw material. So that's why Indonesia is #1 exporting the seaweed, the dry seaweed, I mean. So that's why that is really developed in particular in Eastern Indonesia. I'm also get some information related in Batam, also in several like Bali. That is because the tourist was going down, and then they are starting for the seaweed cultivation, and then they are asking me for develop this method. Then okay, that is for you for seaweed cultivations. Thank you for Ibu Pelita. Thank you very much.
Do I still have time to have another question? It's a short one. But then if there's no time, it's also fine.
Yeah, maybe yeah. There's no time, maybe Mbak Pelita. Maybe in the next session. Okay.
Okay.
Okay. Thank you, Mr. Ma'ruf Kasim.
Thank you very much.
Yeah, thank you for the 3 presenters that's already present their research. We move to the next presenter, Miss Nur Fajriani Nursida. Yeah. Miss Nur Fajriani Nursida.
Yeah, hello.
Yeah. Yeah, please welcome. You can share. Yeah.
Okay, can you see it?
Wait a minute. Yeah, I can see.
Halo.
Yeah.
Oke. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you for the opportunity that has given to me to presenting my research about in vitro antibacterial activity of ethanol extract of sweet potato leaves against Aeromonas hydrophila. First of all, let me introduce myself. My name is Nur Fajriani Nursida. I'm from Institut Teknologi dan Bisnis Maritim Balik-Jiwa based on Makassar, South Sulawesi. The problems of this research is the attack of Aeromonas hydrophila. To overcome the disease, farmers often have to spend more to buy drugs in anticipation of disease. Using the drugs in fish, such as antibiotics, and can lead to resistance to cultivants and the other problems in the environment, so that they are not effective to overcoming the disease. Indonesia have various kind of plants that can increase immunity. One of that is Icomia batata or sweet potato leaves. These sweet potato leaves are the plants that widely available with a relatively short harsh period, and are relatively expensive and rich in nutrients such as carbohydrate, protein, calcium, iron, and vitamins. The vitamins contained in sweet potato leaves are beta carotene, vitamin C, vitamin E, vitamins B, and in addition, sweet potato leaves have polyphenol. As we know that polyphenol are rich with antioxidant. Besides that, this leaf also contains saponin and flavonoid. The sweet potato leaves are also able to increase immunity in tilapia. That is shown from the blood profile, which has a significant difference often give sweet potato leaves by feed formulation. The methods of this research. The first is extract production. The leaves of sweet potato leaves are young leaf, then short and dry. After it pollinate, and the sample is after that the sample is ready to extract. Extraction using method the extraction using a maceration method. The second step is a preparation of Aeromonas hydrophila. The preparation of Aeromonas hydrophila were rejuvenated in nutrient broth and incubated 24 hours in 37 degrees Celsius. The third step is inhibition test. The method for this step is using Kirby-Bauer's method, the diffusion method with paper disc. So the paper disc has been dripped in sweet potato leaf extract as much as 20 microliters. With the first concentration is 5%. The second concentration is 10%, and then 20%, 40%, and the last 80%. With the positive control is tetracycline antibiotics. After that, we can measure the zone diameter. We measure it with a ruler. The diameter zone is measured twice. The result of this study according to Davis 1971, the result of this study, we can see that at 5% extract, it showed that it has a weak inhibition, and in 10 and 20 percent extract, it showed that strong inhibition, and 40 and 80% it showed that a very strong inhibition. My other research about this leaf show a significant recovery to a fish that inject with Aeromonas hydrophylla. The tilapia shown that the lesion getting smaller after dip with this sweet potato extract. Okay, this research was supported by Grant Dosen Formula 2020, Directorate of Research and the RPM, Ministry of Education and Culture Indonesia. Thank you.
Okay, thank you, Miss Nur Fajriani Nursida. Yeah, okay, the next presenter is Miss Karin Octavia Lestari.
Okay, thank you.
Yeah.
I will present my presentation.
Oh, okay.
Thank you very much for the opportunity. I'm here to present my research, which the title is Heavy Metal, Mercury, Cadmium, Lead, and Copper in the Long-Whisker Catfish, Mr. Julio Hamilton, 2020 in Bojonegoro Coastal Waters, Banten Bay, Indonesia, by Karin Octavia Lestari, Sulistiono, and Hefni Effendi from Department of Aquatic Resources Management, Faculty of Fisheries and Marine Science, IPB University, and Environmental Research Center, IPB University, Bogor, Indonesia. A paper presented at the International Conference on Fisheries, Aquatic, and Environmental Sciences on 3rd August 2021, organized by Syah Kuala University. Bojonegoro Coastal Region is a strategic area because it is used as an industrial area, residential and fisheries. Anthropogenic activities such as industrial, domestic, fisheries, and shipping in Bojonegoro are the main contributor to entry of heavy metals into the waters. Heavy metals easily form complex bonds. Heavy metals like mercury, cadmium, lead, and copper in certain level are toxic for organisms. Heavy metals in the water enter the body of the biota through the food chain. Long-whisker catfish are susceptible to exposure to heavy metal because they live at the bottom of water and are tolerant of polluted water. The purpose of this research is to estimating the accumulation of heavy metal in long-whisker catfish meat in Bojonegoro coastal water, Banten Bay, Indonesia. The benefit of the research is to provide information of the accumulation of heavy metal content in long-whisker catfish meat as a basis for managing the Bojonegoro waters, Banten Bay. Sampling of long-whisker catfish was conducted from August until October 2020 in coastal water of Bojonegoro, Banten Province in two sessions. Session 1 is Wadas River and Session 2 is Strate River. The process of dissection and morphometric measurement of long-whisker catfish have been carried out at the Laboratory of Microbiology 1, IPB University. When assaying and analysis of heavy metal content of Mr. Julio carried out at the Agricultural Industry Technology Testing Laboratory, IPB University. Primary data is obtained from heavy metal content of mercury, cadmium, lead, and copper in long-whisker catfish meat with atomic absorption spectrophotometry method. The secondary data is obtained from the content of heavy metal in water and water quality college who researched in the same location in 2020. The content of heavy metal cadmium, according to Surbakti et al. 2021, sampling procedure and AAS analysis. First, long-whisker catfish sampling with purposive sampling, which has previously been studied. A study regarding the habitat of long-whisker catfish in Bojonegara, Banten Bay used 1.5 cast net. Secondly, taking fish meat and measuring leg fish sample were distinguished by two categories, large and small. Large is greater than 10.1 cm and small is less than 10.1 cm. Third is wet description. Use the nitric acid perchloric acid method to separate the fat and solution in the sample. Fourth is analysis of heavy metal using plant atomic absorption spectrophotometry according American Public Health Association 2077. Descriptive analysis is used for compare heavy metal content in long-whisker catfish with National Agency of Drug and Food Control or BPOM, heavy metal content of copper in long-whisker catfish with Directorate General of Drug and Food Control, Ministry of Health, Republic of Indonesia, heavy metal in water and water quality with Republic of Indonesia Government Regulation number 22 of 2021. Quantitative analysis. First is the bioconcentration factor is used to determine the ability of organisms to accumulate heavy metals in waters according to USEPA 2003 and H 1977. The self-limit for consumption is the maximum weight for consuming the organism every week. The determination of the self-limit for consumption can be seen from the smallest limit value of the type of heavy metal content, so that there is no deposition of metal in the body that can cause death. Statistic analysis is using two-factor analysis of variant test without replication or ANOVA using Microsoft Excel 2013. Result and discussion. The content of heavy metal, mercury, cadmium, and lead don't exceed the quality standard according to BPOM 2018. The content of copper in each month doesn't exceed the quality standard according to regulation of the Minister of Health. ANOVA with a 95% confidence interval explain the different size of fish on the heavy metal content of copper did not have a significant effect every month. Copper is one of the essential heavy metals needed by the body of organism. The copper content in the water is also higher when compared to other heavy metals, so that the accumulation of copper in the long-whisker catfish meat has a higher value than the other heavy metal. According to AIDA 2013, copper is one of the heavy metals that function as a blood system hemocyanin and enzymatic formation for aquatic organisms. In the growth stage, fish will actively absorb and use copper obtained from food and in their environment. Bioconcentration factor of copper in small long-whisker catfish on average below to moderate accumulation. This is different from the bioconcentration factor value of large long-whisker catfish, including moderate to high accumulation, reaching 3,120. In addition, the level of mercury, cadmium, and lead accumulation in long-whisker catfish was classified as low. The temperature in October, dissolved oxygen in September and October, and acidity in each month does not exceed the quality standard according to Republic of Indonesia Government Regulation number 22 of 2021. Heavy metal toxicity is affected by a decrease in dissolved oxygen and acidity in waters.
Yeah, Ms. Karin, 2 minutes left.
The content of mercury, cadmium, lead, and copper in water each month does not exceed the quality standard according to Republic of Indonesia Government Regulation number 22 of 2021. The self-limit for consumption is in 1 week of long-whisker catfish meat for adult is in Bojonegara waters, Banten Bay is maximum of 16.03 kilogram of meat per week, while for children can consume much as 4.81 kilogram of meat per week. This value is taken from a heavy metal copper, which has the lowest of MTA value. The conclusion of this research is the content of heavy metal, mercury, cadmium, and lead in long-whiskered catfish was below the detection limit of AAS. While small and large long-whiskered catfish were detected to contain heavy metal copper range 0.699 until 0.831 milligram per kilogram and 2.640 until 10.920 milligram per kilogram. Small-sized long-whiskered catfish bioaccumulate heavy metal copper, including moderate accumulation, and large long-whiskered catfish, including moderate to high accumulation. While the heavy metals, mercury, cadmium, lead, are low accumulation, the self-limit for consumption of long-whiskered catfish meat that we consume is 16.03 kilogram of meat per week for adults, while for children is 4.81 kilogram of meat per week. Suggestion: similar research needs to be carried out at the same location to monitor the heavy metal pollution that occur everywhere. In addition, research of the accumulation of heavy metal, especially in the gills and bill of long-whiskered catfish, need to be done. Thank you very much for the opportunity.
Yeah, thank you, Ms. Karin, for your presentation. Next presenter, please welcome Ms. Dita Fatriani. Ms. Dita. Ms. Dita is already here or not? Yeah, okay, we move to Ms. Hanifah Nedia. Ms. Hanifah Nedia. Ms. Hanifah. Okay. Yeah, we move to Mr. Rizky Hadiwibowo.
Yes.
Yeah, Mr. Rizky. Yeah. Yes, you can.
Can I share our PowerPoint?
Yes.
Okay. So, can you see that our PowerPoint now?
Yeah, Mr. Rizky. Yeah.
Okay. Thank you so much for the committee. So, okay, I will slideshow for our PowerPoint first. Okay. Okay. I do apologize first for the committee if my voice maybe it's unclear because it is raining in Bengkulu now.
Okay, it's okay.
Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay. Good afternoon to all of the participants here, to all of the presenters here. And we also want to say thank you so much to the committee for the opportunity that has given to us to deliver upon our research informations with the title Identifications of the Vibrio bacteria cause fibriosis in spiny lobster, Phanulerus homarus in Bengkulu marine cultivation farm. So as the first author, me, Rizky Hadiwibowo, and also our team from Biology Department of Chemistry, and also collaborate with the Quality Control and Fisheries Product Safety Agency, BKAPM of Bengkulu. So as our introduction, we know that the spiny lobster actually is one of the export commodity that has a very good export high trade value, especially in fishery subsector in Indonesia. But maybe the lobster cultivations business can be failure because some of the infections from the pathogenic bacterium, I mean, such as like a Vibrio subspecies, because this Vibrio subspecies maybe can cause the death of the instar or the larva of the lobster more than 50% in the cultivations. So in our research, very simple. So we just want to know about the kind or maybe the diversity of the Vibrio subspecies that can cause of the fibriosis in the spiny lobster, especially in marine cultivations found in Bengkulu, Indonesia. So as the methodology, we do some of the sample collections of the spiny lobster and do the isolations of the target of the pathogenic bacteria from spiny lobster sample. Actually, some of the particularly from some of the specific organ like gills, stomachs, hepatopancreas, and also hemolymph. And then we grout on the Triptychus soy agar, and also we do the purification of our pathogenic bacteria as a target in TCBS medium. And then after the purifications and the isolations, we identified the pathogenic bacteria by several parameters. First is morphological identification, and also the second one by the biochemical test. And then we make the data analysis. Okay, so from the result and discussion, from the isolations of the target pathogenic bacteria from spiny lobster sample, so over here we can see that the clinical symptoms of the body of spiny lobster that can be infected by the Vibrio from the abdomen, uropod, and also the pleopod, from the gills, and also hepatopancreas. So the character of the spiny lobster infected by the Vibrio, we can see from the red spot of the pleopod, uropod, and also the abdomen. And the color of brownish in the hepatopancreas, and also some of the symptoms in the body surface that can affected by the Vibrio. So from the purification and also from the isolation, we got 5 of the types of the Vibrio as subspecies we successfully isolate from the medium of the TCBS. So this is the figure 2, the bacterial isolate from spiny lobster organ. We got on the TSA, Triptychus soy agar, that we have been isolated from several organs, from hemolymphs, stomach, gill, hepatopancreas, and then we give the code SI1, US2, IN3, and also HE2, and also HE3. So based on the morphology identification of the pathogenic bacteria, from 5 of the pathogenic bacteria that we successfully grout on the TCBS or maybe in the TSA medium, we can see that bacteria over here from the color, margin, elevations, and form. So we can see here from 5 of the pathogenic bacteria, they have a similar character in the morphology. And then based on the biochemical test of the pathogenic bacteria that we have done, we do the assay of the biochemical test from the Mayo test, triple sugar iron agar, Simon citrate agar, urease test, lysine iron agar, and oxidative fermentative test. So from this data, we can see that over here, from this data, we can see the differences, and maybe there's some of the variant result from the biochemical test. So from the morphological and also the biochemical test, we can conclude that based on the Berge's Manual of Systematic Bacteriological books, and also from the Austin and Austin in 2012, from the morphological observation and biochemical test characteristic, we can see that the isolate of XI1, bacterial isolate, were close related with Vibrio caricae. The second isolate of IN3 were close related with Vibrio anguillarum. And then UST2 and also USG2 bacterial were close related with Vibrio orderi. And also the bacterial isolate of HE2 were close related with Vibrio splendidus, and the other isolate, HE3 isolate, close related with Vibrio alginolithicus. So from the 5 of the bacteria that we have find, 5 of them are Vibrio. And then the acknowledgement, the author would like to thank to LPPM Universitas Bengkulu from the Hibah Penelitian Fundamental Unit, and also to BKIPM of Bengkulu and all of the parties that have been already helped for the completion of the research. Maybe only that, the data of the informations that we have informed to all of the participants here. Thank you so much, and we bring back to the moderator. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Okay, thank you, Mr. Rizky Hadiwibowo, for the interesting research. Are there any questions for Ms. Nur Fadriani, Ms. Karin, and Mr. Rizky Hadiwibowo? Yeah? You may ask question for them. Okay, if there's no questions, we can move to the next presenter. Yeah? No questions?
I do have a short question.
Okay, Ms. Pelita.
Is it okay?
Yes,
Okay.
it's okay.
It's only one question. It's about the heavy metal thing. I mean, in her result there, I'm sorry, I don't remember the name of the presenter.
Miss Karin.
Yeah, Miss Karin. Because there's two of them who should be present about the heavy metal, right? So the Miss Karin, your results show that there is no difference between the large size of fish and then the small one. So it can be said that the heavy metal contain in their meat is the same. But then my question is there any relation between the concentration of heavy metal in the environment and then the one that accumulate in the fish body? For example, the highest concentration environment mean also high high concentration in the meat, for example. And then can you suggest, for example, if it's not different between the large and the small one, is it more good to eat the big fish rather than small fish? Because then if we eat small fish, definitely we consume more heavy metal. Is it the conclusion like it?
Okay, yeah. There are two questions for Ms. Karin. Do you want to answer now?
Okay, thank you for the question, Ms. Pelita. Yes, this is this is anthropogenic activities in this water will lead to greater waste input of course, this condition will an impact on the waters and biota. And small categories and big categories in this research is I don't hear very simply.
So you want me to repeat my question?
Yeah,
Simple
yeah.
way?
Yeah.
So if we found a high concentration of heavy metal in environment, is it also mean that we will found high concentration in fish?
Yeah.
Oh yeah. In environment, it's which is a high concentration of heavy metal, we can know that the accumulation of heavy metal is high. This is represent on my research, because in my research is the height of accumulation is copper, and in the fish is the high accumulation is copper, so it can be determined. Thank you.
Yeah. So it have a relationship, yeah.
Yes, it has a relationship.
For the second question, Ms. Karin, about the size of a fish, yeah.
About the size of the fish is we can consume according to the MTI or MTI is the white. MTI, this value is taken from the heavy metal, is the lowest value of the all of the copper in all of the heavy metal in this research.
Okay,
So
thank you.
we can conclude that small fish is more edible than big fish if it have contain heavy metal.
Okay. So it will be safe if we consume the smaller fish, yeah, Ms. Karin?
Yes.
Okay,
Yes.
Thank you, Ms. Karin, for your explanation. Yeah, are there any questions? Yeah. Okay, maybe we move to the next presenter, Mr. Iksan. Yeah, Mr. Iksan. Okay. The next one, Miss Ita Apriani.
Yeah.
Yeah. Okay, Miss Ita, do you want to share?
Yeah.
Yeah.
Thank you. Okay, you can see this myself. Yes. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good evening to all committee and seminar participants. My name is Ita Apriani. I'm from Academy Perikanan Yogyakarta. Today, I will present with my title Proxima Test of Meat Catfish, Clarias gariepinus, cultivated with biofloc system. Okay, in introduction, high market demand makes catfish farming technology continues to develop. One of them is the application of the technology is biofloc system. And then in my studies, I am to determine the result of the Proxima test of catfish meat culture with a biofloc system using different type of probiotic bacteria. My research method, the method used was a completely randomized design with 4 treatments and 3 replication, namely P1 as control, P2 probiotic nitrobacter, P3 probiotic lactobacillus, and P4 probiotic bacillus. Fish were reared for 300 days feeding time, 3 days as much as 5%, adding probiotic every week as much as 10 milliliters. The data from the Proxima test were analyzed descriptively, covering the content of protein, lipid, water content, and extract without nitrogen as a backend. The result and discussion table of result of Proxima analysis of catfish meat for treatment and as protein, lipid, crude, fiber, and BTN in dry weight. The experimental meat in P2 had the highest protein content and BTN the lowest compared to other treatments. Treatment P3 had the highest lipid content and the lowest ash content. And the treatment P4 had the lowest protein and lipid and the highest content for ash and BTN. Nitrobacter bacteria in water is thought to help improve water quality and increase the number of microbes in the intestine so that fish are able to absorb nutrients better and produce quality fish meat. And Bacillus subtilis in water better growth performance and modifies the intestinal microbiota, reducing the class of potential pathogen agent species. Microorganism population may improve host health. The conclusion of this study is that the addition of probiotic bacteria affect the result of the Proxima test of meat. The addition of Nitrobacter bacteria increased protein level. The addition of Lactobacillus increased lipid content, and the addition of Bacillus subtilis increased the mineral content in the meat of catfish. Okay, I think that's all from me. Thank you so much, and I give back to the moderator. Thank you.
Okay, thank you, Ms. Ita Apriani, for your presentation. So the next presenter is Ms. Pelita Torina. Okay. Yeah, please welcome.
It's already me. Okay. I will share my screen here. Can you see it?
Yeah, Mbak Pelita.
Okay, thank you. Okay, let's start. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good day, everybody. My name is Pelita Oktorina from Sukabumi West Java, and my talk today is about phytoremediation in Sandpit Lake. And this is a part of our project in managing Sandpit Lake in Sukabumi Regency, done by me and my colleague from Department of Aquaculture, Muhammadiyah University of Sukabumi. Wait. Oh, my slide do not move, right?
Yes.
Okay then.
But it's already.
It's okay now?
No.
Okay,
Yes.
thank you. So at the beginning, I will speak a little about Sandpit Lake. It's a small man-made lake located near the urban area, therefore it's so fragile to the anthropogenic activity. And Sandpit Lake in Sukabumi Regency also has this criteria where the size range between 2 until 5 hectares, and then with 5 to 20 meter deep. And then it has a less littoral area, or let's say less beach. Moreover, but they have a potential to be developed as aquaculture site, but their trophic status, apparently recognized as mesotrophic and eutrophic. And then regarding this topic status, if we want to apply an aquaculture in this area, it should be an eco-friendly aquaculture. And then the next, we want to introduce a system called vegetable floating raft, or VFR, as a phytoremediation tools. The reason why we want to apply this the VFR in Sandpit Lake were, first, we know that macrophyte is a nutrient absorber, and the ability in enhanced water quality was proven in many studies. In Sandpit Lake, using submerged macrophyte is not an option because I already told you before, the littoral area is not large, and then the sediment mainly still contains sand or big rock, so it's not suitable for plant. And using floating macrophyte also risky regarding the possibility of blooming, because we have experienced another Sandpit Lake is actually covered by I don't know the English word, it's etching gondok. And then for all the reason we interest in planting vegetable in Sandpit Lake on a floating raft with a hydroponic system as the main principle. So we hope it will serve as a floating macrophyte and avoiding possibility of blooming. And also we can take benefit from the vegetable production. But then we come up with a big question. Can it work? Because we kind of have little literature about it, and then there's also we cannot find also another study regarding this, so it's really big question. Therefore, we tested the effectivity of VFR in reducing nutrient from the aquaculture system, and then to get this, we use the combination of floating cage fish and vegetable floating raft in aquaponic, and we did it in a mesocosm. And then for the methods, for the hydroponic system, we use lettuce as the vegetable, and then we have for treatment of VFR coverage. We use the coverage of VFR, which were 0%, 20%, 40%, and 60% of the mesocosm or mesocosm area. We assume this mesocosm as a fish cage. So, and then for the aquaculture system, we use 25 fingerling fish with size 10 to 12 centimeters. It's a tilapia fish, and then we fed the fish with artificial fish pellet with 40% from the fish biomass, and we fed them twice a day. And then for the mesocosm, we use 12 mesocosm because we have a triplicate. And then we conducted the experiment for 7 days. And for the data, we collected water parameters such as temperature, pH, DO, and then the nutrients were TAN, nitrate, nitrate, and orthophosphate. And then we also collected the data about lettuce biomass, superfire rate, and then the growth rate. And then we collected the data 2 times. Is the day 0, or the first day we conducted the experiment, and then the last day is the day 7. And then we ran the ANOVA and then the t-test. And then this is our mesocosm design. So as you see here, this is actually a floating cage, or in Indonesian language we call it jaring apung. So it's exactly similar with karamba jaring apung, but then we replace the cage net with plastic, so as to be to serve as the mesocosm. And that this is the view from the upper side, and then this is from the side view. We will see something like this, and then this is what the originally picture that we have during the experiment. And then for the VFR, or the vegetable floating raft design, we have something like this. This is the actually photos of our VFR, and then this is the design. So this is the bamboos, this is the plastic bottle, because as a boy, because it's cheap, And then, so basically this is a cheap way to have a hydroponic in lake. So the result, apparently the result, we found that dissolved oxygen level decrease in all mesocosm. That's not what we're expecting, of course. And then the nutrient level generally will increase. Meanwhile, pH and temperature remain stable. As you see here, and then, and then the lettuce biomass also decreased. So can be said that the lettuce do not, probably not survive in this environment. And then we found it's due to the negative growth rate. And even though from the graph we saw variation, you see here, there's a variation between treatment. They are not really the same. But then when we test it statistically, the difference between treatment are not significant. So, and then we run another analysis, and then we plotting the change of water parameter with the treatment to see the treatment effect. Again, we couldn't see the relationship between treatment and water parameter change or several nutrient change, except the nitrate. As we see here, even though we see that the change of oxygen is really in the treatment B is lower than the other, but then statistically it's not significant. And then for this one, the nitrate, what I told you before, there's a tendency, or there is a like a pattern that the concentration of nitrate relate to the size of the coverage, the VFR coverage. Larger coverage mean we will have larger or a high nitrate concentration. So, and then another is about the biomass. We see the relationship between the biomass and the size of VFR, where the larger VFR, the biomass will reduce more than the other. So it's quite disappointed because we thought larger FVR will resulting high biomass, but then what happened is the opposite of it. And then, but then we also see that there was a tendency of coverage, 40% had a better lettuce survival rate rather than the other one, even though it's only tendency, it's not really significant statistically. So maybe if we run another model, we will be able to identify the significance. Oh, sorry, so for the conclusion, even though we found the not significant difference between treatment, means that there is no proof which coverage is the best, but I think we will continue with the 40% coverage to go to the further experiment. And then we also identified, we also recognize that there should be a further works which were study regarding vegetable species and age, and also about the plantation medium. So thank you for listening.
Yeah, thank you, Ms. Pelita. It's very interesting about aquaponic. Yeah, okay, the next presenter I will call again Mr. Ihsan. I think the representative is already here. Yeah, the representative of Mr. Ihsan Research. Sri Mulyani. Miss Sri Mulyani, please welcome. Okay. Okay. Okay, it's okay. So we move to another person. Miss Nur Indah Sari Arbit. Miss Nur Indah Sari.
Assalamualaikum.
Ya, waalaikumsalam ya, Miss Nur.
Can I say?
Yeah. Do you want to share the presentation? Okay, Miss Norinda.
Yes, it's wait a minute.
Yeah.
Yes, can you see my presentation?
Not yet.
Okay. How about this?
No. Maybe you can stop and share again, maybe.
Okay.
Ada muncul.
Yeah,
Yeah,
bagus.
we cannot see your PPT.
Okay. Wait a minute.
Because we don't have also your files.
Yes.
Okay.
Okay. I will try. To
Okay.
share it.
You want to try later, maybe? Yeah.
Maybe can share the PPT to moderator and moderator start.
Yeah, but we don't have the files. Yeah. Okay.
Okay, I will share my
Okay, it's okay. Maybe you can present later. Yeah, Miss Norinda, you can share to our committee. And now we move to the next presenter, Mr. Muklisin, or the representative for his research. Mr. Ramadi. Yeah, Mr. Ramadi. Yes, welcome. You can share. Do you want to share your PPT? Yeah, or I have to share it. Okay. Yeah.
My name is Rahmadi from Department of Aquaculture, Faculty of Marine and Fisheries, University of Kuala.
Okay, maybe the volume is very low.
Today,
you can raise your volume.
Today, my presentation about embryogenesis of climbing birds Anabas testimonius at incubation.
Sorry, Mr. Ramadi, you can raise more your volume. Yeah. Okay. You want to try again?
Hello?
Yeah, it's better.
Okay, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
My name is Rahmadi from Department of Aquaculture, Faculty of Marine and Fisheries, University of Kuala. Today, my presentation about embryogenesis of climbing birds Anabas testimonius at incubation temperature of 28 degrees Celsius. Introduction. Climbing birds Anabas testimonius, or locally named Betok, is a freshwater fish that has economic value and has the potential to be developed as a target for commercial aquaculture. Studies on the climbing bird fish have been carried out by several researchers, for instance by biological, stocking density, growth, and spawning. Embryology development in cultured fish is important and evident in aquacultural practice and fish production. However, embryonic development of climbing birds Anabastes styreneus has never been carried out. This information is crucial for understanding of the early history of fish and useful for breeding purposes. To observe the process of embryogenesis is climbing bird Anabastes styreneus. Material and method. The study was conducted from March to April 2021 at Fish Hatchery Laboratory, Faculty of Marine and Fisheries. Embryogenesis observation. The embryogenesis process was observed by randomly taken several eggs from the incubation aquarium every 15 minutes and then placing the egg in the object container and observing them using a microscope. During the observation of embryogenesis, the picture of embryonic development and then duration of every phase of embryonic development were recorded. Data analysis, data and the phase and time of embryonic development for each phase were presented in figure and then analyzed descriptively. Result, the embryogenesis of the climbing bird fish is divided into 6 phases, namely by the clophage, morula, blastula, gastrula, organogenesis, and hatching. As can be seen on this slide, this is step-by-step embryogenesis Anabastes styreneus. In terms of incubation time, the egg of Anabastes styreneus, which were incubated at 28 degrees Celsius, hatched within 18 hours and 38 minutes after the fertilization. At the same incubation temperature towers, egg hatch after 18 hours and 19 minutes of fertilization. Conclusion. The embryogenesis of Anabastes styreneus at an incubation temperature of 28 degrees Celsius is divided into 6 phases, namely by clophage, which lasts for 3 hours, 5 minutes, morula lasts 4 hours, 30 minutes, blastula for 5 hours, 40 minutes, blastula for 8 hours, 47 minutes, organogenesis for 15 hours, 39 minutes, and hatching 18 hours, 38 minutes after fertilization. Thank you for attention.
Okay, thank you, Mr. Ramadi, for your presentation. Okay, so for the 3 presenters, are there any questions for them? Miss Ita, Miss Pelita, and Mr. Ramadi. Okay, are there any questions? Yeah. Miss Pelita, I think Dr. Martin already chat you. It's about the excellent use of ggplot for the results. Maybe you already see the chat.
Yeah,
Okay.
thank you. Thank you for the compliment, but yeah, it's not my best.
Yeah.
Need to learn more.
A very nice presentation. Thank you.
Yeah, I would like to learn more from you, Martin, if you're okay. I mean,
Please email me.
okay, I will.
Okay. Yeah.
But then it seems like I always deliver question. If it is okay, I also have another one because I'm so interesting for all the
okay.
theme. I mean,
Yeah, it's okay, Miss Pelita. You can answer.
it's the question about the embryogenesis is really interesting for me.
Yeah, Mr. Ramadi.
Because,
Yeah.
but then I'm not really get the conclusion. Is it you try to say that the temperature increase the time during the embryogenesis? It means that more heat means that fish will hatch faster. Is it that the conclusion?
Yeah, Mr. Ramadi. What is your conclusion? Maybe you can share to us. Yeah, Mr. Ramadi, you can share the conclusion maybe. Yeah.
I think Mr. Ramadi is still mute.
Mr. Ramadi, you are still mute. Maybe you can share the conclusion for us. Yeah, you can share the conclusion.
Yes.
Miss Pelita asked about the conclusion. Maybe you can read again.
Okay, Miss. Only focus temperature of 28 degrees Celsius.
Yeah.
So these temperatures really make the embryogenesis faster. The 28 degree Celsius, or
yeah. Mr. Ramadi, you use 28 degrees Celsius, yeah, for this research.
Yes.
Okay. Okay, you will win. Yeah, Mr. Ramadi. It's okay. Yeah, maybe,
sorry, because
yeah.
this is my partner in research, so I will have him to answer this question from Miss Pelita, right?
Okay.
Yeah.
So our research is focused on 20 degrees Celsius, and we don't compare with another temperature. So I'm not sure another temperature will be make more faster or slower. We need still to do research to evaluate that. Thank you very much.
Okay, clear. Okay, clear. Thank you.
Yeah, on the temperature. Okay, thank you, Mr. Ramadi and your team. Okay, the next one. Is there any questions? Okay. Maybe we can. I will call again. Miss Nur Indah, are you ready now?
Yes.
Okay. Yeah. Do you want to share again?
Yeah. Let me to share.
Indah, sorry. Yeah. Oh, with your handphone?
Yeah. Sorry to use my handphone because the signal.
Okay. Yeah, it's okay.
Okay.
Yeah. Okay, we can see now.
Yes.
Hello.
Ya, halo.
Ya.
Ya. Baik. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good day, all of the participants. Thank you very much for opportunity to give me an opportunity to present my presentation, and the title is about growth and carrageenan content of seaweed Capapicus alvarezi cultivated at different depths using the bag method. Introduction. The waters of West Sulawesi have the potential for the development of seaweed cultivation. The clear waters making it possible not only to use the surface of the water for seaweed cultivation, also the problem in these waters is the current, which is quite heavy, and there are still many natural predators such as baronang fish, turtle, and several other types of fish. So the objective of this study to determine the effect of depth and the growth, the carrageenan content of Capapicus alvarezi using bed. This is the material and method. The research was conducted on Batua Island, Tonyaman Village, Polewali Mandar District, West Sulawesi Province. And the seaweed sample used in this study was Capapicus alvarezi, and parameter observed is growth and carrageenan content. You can look the method. It is we use a bag for the seaweed. So the study content of for treatments and 3 replication seaweed in bags was hooked vertically at different cultivation depth. In P1 is 25 centimeter, P2 50 centimeter, P3 75 centimeter, and P4 100 centimeter. And in each depth is using bag. It is I think very inspiring Vertinet from Pak Ma'ruf Kasim. And result discussion the results so that the significantly different from growth seaweed cultivation at depth P1 saw a specific growth and 6.57% higher than P4 treatment with a specific growth 3.14%. And for the carrageenan content, a depth of 25 centimeter is 25.59%, and the higher content is at a depth of 25 centimeter as much as 34.51%. The conclusion from this research is that the highest carrageenan content is obtained from a depth of 75 centimeter below sea level to obtain optimal growth and carrageenan content of Capapicus alvarezi on Batua Island should be planned at a depth of 25 centimeter until 75 centimeter below the water surface. Thank you.
Okay, thank you, Ms. Norinda, for your presentation. Next presenter, Ms. Dini Islama, or the representative.
Yes, I would like to
Mr.
present.
Agung.
Yeah.
Mr. Agung.
For the PPT, a moderator to share or me to share?
If you have problem, I will share your PPT.
Can you see? Yeah,
we can see. Yeah.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Everybody, good afternoon. My honorable to moderator, Mrs. K, and all participant and all presenter. In this opportunity, I would like to present our research about supplementation of gamma leaf floor in commercial feed on the growth and feed conversion ratio of Nirwana tilapia of Oreopramis nauticus fingerling. In Indonesia, tilapia is classified as one of the leading aquaculture commodities and its production continues to be increased. Tilapia production increased every year with an average increase in the amount of production reaching 31% in 2013 and until 2017. Research on the supplementation of gamma leaf floor in commercial feed on the growth and feed conversion ratio of Nirwana tilapia fingerling has not been reported. The study aimed to examine the effect of supplementation of gamma leaf floor in commercial feed on the growth and feed conversion ratio of Nirwana tilapia fingerling. And then material and method, time, location, and experimental design. This research was conducted from April to May 2019 at the Laboratory of Seed and Fish Feed Production, Polytechnic of Indonesian Venezuela in the Aceh Besar district. The experimental design used was a non-factorial, completely randomized design consisting of 4 treatment and 4 replication. Preparation and production of feed test. The feed used in the study was commercial pellet with low protein content. 28% of protein content. The fermented gamma leaf flour is mixed with commercial pellet that had been grown into flour. Feed was given as much as 5% of the weight of the Nirwana tilapia fingerling biomass. And then maintenance of Nirwana tilapia. Nirwana tilapia seed stocking density is 1 individual per liter. The volume of water filled in the container is 40 liter. Nirwana tilapia fingerling maintenance period lasts for 40 days. Parameter and data analysis. In the study, the parameter measured include weight gain, length gain, daily growth rate, feed conversion ratio, survival rate. Data were analysis using analysis of variant or ANOVA in the SPSS version 22 software. The result of analysis show that the gamma leaf flour supplementation in commercial feed gave a significant effect on weight gain, length gain, daily growth rate, and feed conversion ratio, but did not give a significant effect on survival rate, Nirwana tilapia fingerling. Can you see that in the table? We result in the study give a significant effect. The result of the feed proximate test showed that the protein content was higher in the treatment of gamma leaf flour supplementation in the commercial feed than in the control treatment. Conclusion, supplementation of gamma leaf flour in commercial feed gave a significant effect on weight gain, length gain, daily growth rate, and feed conversion ratio, but did not give a significant effect on the survival rate of Nirwana tilapia fingerling. The best dose to increase the growth and feed conversion ratio of Nirwana tilapia fingerling was commercial feed with supplementation of gamma leaf flour in the 10% per kilo feed. Thank you.
Okay,
I getting back to the moderator.
Yeah, thank you, Mr. Agung. Okay, the next presenter is Miss Umi Mardia.
Okay, thank you very much. Are you hear my voice?
Yes, Miss Umi.
Okay, thank
Yeah.
you, Mrs. Iska.
Okay. Yeah, you can see the PPT. Yeah.
Yes, okay, thank you. Please, full screen. Okay, thank you very much. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. An honor to meet you here by virtual conference. Let me introduce myself. My name is Umi Mardia Batubara, and I'm from Riau University, Pekanbaru. In this presentation, I would like to present the result of our research entitled Optimization of an Effective Ground Medium for Biomass Production of Bacillus cereus. Next. Okay, this is our team. From left, Mr. Feliatra, it's me. In medium, Mr. Irwan and Mrs. Adelina. We are from a member at Laboratory of Marine Science, Marine Microbiology Department of Marine Science, Fisheries and Marine Science Faculty, and Mrs. Adelina is a member at Laboratory of Aquaculture, Department of Aquaculture, Fisheries and Marine Science Faculty. Next. Okay, introduction. Feed or feed fish is the main nutrient needed in aquaculture. Feed nutrient contain complex compounds such as proteins, carbohydrates, lipids, vitamins, water, and minerals. The protein composition is in feed formulation account for 16% of all components in the feed. However, the high protein requirement is complicated by the high price of conventional protein. Therefore, it is necessary to find an alternative protein sources derived from microorganisms. One of them is by utilizing protein derived from Bacillus cereus SN7 and consortium of Bacillus cereus. So, why we choose Bacillus cereus SN7 and consortium of Bacillus cereus? This is based on the result of please back to yes, okay. This is based on the result of previous studies on the potential of Bacillus cereus as a candidate for probiotic bacteria. These are our publication paper about the potential of Bacillus cereus and consortium of Bacillus cereus. In previous research, we get potential from marine heterotrophic, and in the right you can see many strands from Bacillus cereus, and 1, 2, 3, 4, 5 strand we use in this research. Okay, next. So, to develop the potential of all strands of Bacillus cereus as single-cell protein, an effective or an efficient ground medium is needed, and one of them by utilizing the industrial waste. One of them is a tofu liquid waste. Okay, there are many reasons why we choose tofu liquid waste as the ground medium for Bacillus cereus. First, tofu liquid waste is an environmental problem caused by tofu industries. Second, the tofu liquid waste contain very high organic substances and abundant in nature. So we assume that the utilization of tofu liquid waste is expected to increase Bacillus cereus biomass, which will be used as a substitute protein source in fish feed. So this study aims to determine the effectiveness of tofu liquid waste as a ground medium for biomass production of Bacillus cereus as a single cell protein. Okay, next. Methodology. This study used an experimental method. The stage of this research are bacterial strain. The bacterial used in this study consists of two parts, namely Bacillus cereus SN7 is a single strain, and Bacillus consortium is a combination of 5 strain. Bacillus cereus SN7, Bacillus cereus SP4, Bacillus cereus S5, Bacillus cereus X MB051, and Bacillus cereus B52. All bacterial strains were obtained from the collection of the Laboratory of Marine Microbiology, Department of Marine Science, Faculty of Fisheries and Marine Science, Universitas Riau. For culture media and ground condition, all bacteria were grown on modified media containing tofu liquid waste with different concentration, such as 8%, 10%, and 12%, enriched with 10% skim milk, butter phosphate, and vitamin B12. The mixture medium was dissolved in distilled water and put in a 120 milliglass bottle, and then Bacillus cereus SN7 and Bacillus cereus consortium were subsequently cultured in ground media that had been sterilized by autoclaving at 121 degrees Celsius for 15 minutes. And furthermore, these were incubated overnight at 37 degrees Celsius, were performed under aerobic condition on a water bath shaker at 115 revolutions per minute. For analytical methods, we use three analytical methods to know what the effect of tofu liquid waste. The first is total plate count, second is cell biomass, and third is cell dry weight. Okay, and you can look the method from my slide. Okay, next, Miss Fiska. Result and discussion. For the first, we got the information about microbial biomass production, and we can look based on the data in table 1. It can be seen that the optical density value of Bacillus cereus SN7 and Bacillus cereus consortium were highest in ground media with a concentration of 12%. It significantly an increase in the absorbance value was seen at the incubation period at 6 and 12 hours. Okay, next. On the table 2, measurement of cell dry weight of Bacillus cereus SN7 and Bacillus cereus consortium, we can see the highest measurement of cell dry weight also show in ground media with a concentration of 12%, and it's significantly the highest amount of biomass was also seen at incubation period 6 and 12 hours. Okay, next slide, please. Yes. Same with data on table 1 and 2. The data on the table 3 show that the number of viable cells of Bacillus cereus SN7 and Bacillus cereus consortium were highest in ground media with a concentration of 12%. And it's significantly that increase in the absorbance value was seen at the incubation period 6 and 12 hours. Just Because the third result show the same potential for increasing biomass, it can be said the tofu liquid waste can be used as a medium for bacterial growth in the production of single-cell protein of Bacillus cereus. Okay, next. Okay, based on the data obtained, it also shows that Bacillus cereus SN7 and the Bacillus cereus consortium both have the ability to grow in tofu liquid waste. The result on the analysis show that this causes by many factors, including the nutrient content in tofu wastewater are very complex. Tofu liquid waste modified media content the appropriate nutritional composition need for Bacillus cereus SN7 and Bacillus cereus consortium as a single cell protein. And about 16% of the composition of tofu wastewater or liquid water contain protein, which is degraded into organic compound and is useful as a macronutrient and micronutrient for heterotrophic bacteria. And the last reason why tofu liquid waste can use the medium for Bacillus cereus is skim milk. Skim milk is used as an additional source of nitrogen because it contains lactose, casein, and other nutrients needed for bacterial growth. And from the data, it is also seen that the bacterial growth curve form include the lag phase or adaptation phase and exponential phase, stationary phase, and death phase. Microbial growth curve can provide useful information for understanding microbial growth and selecting optimal growth. Okay. Conclusion. Based on the result and discussion, it can be concluded that tofu liquid waste could be used as an alternative grown medium for Bacillus cereus SN7 and Bacillus cereus consortium. And the best modified media formulation for Bacillus cereus SN7 and Bacillus cereus consortium grown was tofu liquid waste at a concentration of 12% enriched with 10% skim milk. Cell biomass of Bacillus cereus SN7 and Bacillus cereus consortium increased significantly at 6 to 12 hours. Acknowledgement. The team would like to thank to Institute for Research and Community Services, Universitas Riau, and the Ministry of Research and Technology Indonesia, or now we call Kemendikbud. Yeah. Well, thank you very much for your kind attention, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Umi, for your presentation. Okay, maybe before we move to the answer and question session, I will call again the last one, Miss Sri Mulyani. Okay, Miss Sri Mulyani.
Okay.
Yeah. Can you share the PPT? Yeah. Okay. Yeah. Yeah, okay.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Going to presented by analysis of heavy metal content of PBN lead engine and zinc in Pacific oyster cultivation in Alunaga village, Banda Aceh. Alunaga region is one area that is used as cultivation land, especially Pacific oyster farming land. The type of oyster is cultivated by the local community is Crassus triagygus oyster. Crassus triagygus is the dominant type of oyster in the coastal area of Aceh. This type of lead and zinc have potential pollution of water due to the loading and unloading activities of fishery product around the oyster cultivation area. According to Isan et al. 2020, oyster cultivation developed in the Tibang Alunaga Reservoir has the best growth and survival of broiler oyster, which is found in treatment with a density of 25 in per 1,500 centimeters. However, research on the analysis of heavy metal content in oyster cultivation developed in surface containers has never been carried out. So, this research is important to be obtained information on how much heavy metal content of lead and zinc is contaminated in oyster on surface container. Material and methods, location and time of research. The research conducted in June 2020, the sampling of oyster is collected at Alunaga village, Banda Aceh, of Aceh province. The analysis of heavy metal, lead and zinc will be conducted at the Aceh Industrial Standardization of Research Agency, Baristan Laboratory. The location is in figure 3.1. Okay, next. Research procedure. The determination of research point, the sampling was conducted at the research location in a cultivation container of the surface. The method used in this research is using sample random sampling. The sampling is carried out directly on the cultivation container so that the sample will have similar opportunity to be a sample that can represent the population. Data analysis. The analysis data in this study is data that come from the AAS tool. The data will be present in table and graph that will determine the value of observance and the concentration of lead and zinc. The regression concentration was obtained based on the regression value of the calibration curve, Supriyatno and Lely Fajri, 2009. The formula used to determine the level of metal, lead, and zinc are alias the following. Not track regression concentration, P dilution factor, V solvent volume, H weight sample volume. Result. Based on the result of the analysis concentration of heavy metal, lead, and zinc in oyster-made Pacific region, which were analyzed using the AAS tool, are presented in table 4.1. Table 4.1, the result of the analysis of metal concentration, lead and zinc in Pacific oyster. You can locate. Okay, next. Discussion. This study showed that the level of heavy metal pollution at the oyster cultivation location is caused by the mining activities that produce waste. Based on the result of the observation of like heavy metal contamination in Pacific oyster, in the three treatment result were not detected in the study. Of sample analysis result for PB metal for lead metal showed the same result. This proved that the oyster grown in container on the surface at the study site area not contaminated by this type of heavy metal. In the observation of zinc heavy metal contamination in Pacific oyster, in the tree treatment, it was found that treatment was
Okay, 2 minutes left.
Okay. Heavy metal and divided into type, namely essential and non-essential heavy metal. The first type is an essential heavy metal that is needed by every living organism, but some of them to a certain degree to area toxic. Generally, people at the research location are still looking for oyster using the old method of swimming and diving. However, this method still has risks, so the idea arose to conduct research on oyster cultivation developed in container on the surface of called the sinking raft method. According to Zainura et al. 2016, there are several methods for oyster cultivation, such as the hang method, the rack method, and the lying method and rock substrates. Okay. Conclusion. Based on the result of the study, is found that the heavy metal content zinc in the Pacific oyster cultivate in surface container in Alunaga village, Banda Aceh City, with the high contamination. This value Icfaes quality standard reason. Meanwhile, the heavy metal content lead in Pacific oyster and not detected by the AAS tool. So this Pacific oyster were safe for consumption. Okay, thank you.
Yeah, thank you, Ms. Sri Mulyani. Okay, are there any questions for presenters, Ms. Nur Indah, Mr. Agung, Ms. Umi, and Ms. Sri Mulyani? Yeah. Okay. Yeah, I think. Yeah, Miss Irma, do you want to ask question? Yeah.
Okay, thank you for the time, for giving me the time for ask the question. I have one question for Dr. Nur Indah, yeah, which the title growth and current content, blah blah blah. Okay, my question is, what is the main parameter that affect the seaweed growth in the West Sulawesi that you mentioned before in your study area? And then the second question is about you use the bag method for cultivate the seaweed. You have the reference. What is the advantage from this method if we compare with the line method or the other method for the cultivate the seaweed? I think just two questions, Mrs. Khan. Thank you.
Thank you, Ms. Irma, for the questions. Okay, Ms. Nur Indah, it's already here. Ms. Nur Indah, there are two questions for there are two questions for Ms. Nur Indah.
Sorry, can you repeat again? Because
Yeah, no problem.
I
Very
have
interesting
already
topic that you
applied.
mentioned before. I have two questions for you.
Yes.
The first question is about the main parameter that affect the seaweed growth and West Sulawesi that you have mentioned before. And then the second is about the advantage of the using bag method if we compare with the another method like the line method or other method for the cultivate. Just two questions.
Yes. Thank you very much for the question. The main parameter I use for growth and keraginan. So I want to see that the growth and the keraginan that if we use the depth and a different depth and we use a bag and advantage if we use a bag we it can avoid the predator and it can make the seaweed not growth very well. So we use the bag because in Tonyaman Island it's very cleaning it's cleaning so the for the like ice is more, there is no we can find it but the predator there is a lot of there so that we use the bag like that.
Okay, thank you, Ms. Nur Indah, for the explanation about the questions. So now we are in the end of the session for oral presentation. Thank you very much for all presentation. It was very great presentation, great research, and also thank you very much for Dr. Martin Wilkes, for joining with us until now. And
my pleasure. Thank you.
Okay, Dr. Martin.
Thank you. Thank you very much.
Okay, thank you. After this session, we have another session, poster session, but before that, we will take a break about 10 minutes and we can join again. Okay, thank you very much for...
Thank you very much, Bu Siska.
Okay, don't forget to join
Thank you, Bu Siska.
the closing ceremony, yeah?
Thank you, Pak Agung. You looking fat.
Thank you, Pak Dedi.
Kamu please.
So the closing ceremony is at 15:10. Okay. Don't miss it. Yes.
Nulisin masih ada ya?
Masih Pak. Sepertinya masih.
Kayaknya promosi itu mana? Promosi masih sibuk ini, mempersiapkan penutupannya. Ini kita pindah ya, Pak Dedi, pindah itu ya, pindah link ya.
Boleh.
Kita pindah link Pak, mungkin setelah poster nanti ya.
Oh, kita lihat poster dulu ya.
Boleh join poster.
Boleh join poster Pak di room ini ya. We will be having a poster presentation after this oral presentation. Prof. Ma'ruf. Please join us. Feel free. Bu Siska, when the poster presentation started? Bu Irma?
Ya, Pak Dedi.
What time the poster presentation started?
We have break for 10 minutes. After
10
that,
minutes.
we come back again for poster session. Just for
Okay,
10
thank
minutes
you.
for our prayer.
Okay, thank you, Bu Irma.
Thank you. Welcome.
Okay, good evening everybody. We will start again the poster session. Before we start this session, I would like to explain some presentation rules for the poster session. So the time limit for the poster presentation is only 5 minutes, and it's only flash presentation without questions. Okay. Yeah, it's only 5 minutes and without questions. So for wait. Okay, I think we will start this poster session now. Yeah. Okay. I will call the first presenter for poster session is Miss Iko Imelda Arisa. Yeah, Miss Iko already here? Yeah. Okay, I will share the the poster. Yeah. Wait a minute. Okay. Yeah. Okay, please welcome Miss Iko to present.
Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, everyone. Thank you for your attention. My name is Iko Imelda Arisa. I'm from Aquaculture Department, Marine and Fisheries Faculty, Universitas Syah Kuala. Today, I want to present our research about the effect Calotropis gigantea leaf extract on egg stability and survival of Barbonimos goniotus larvae. This study aims to determine the Calotropis gigantea extract again stability of Barbonimos goniotus eggs infected by pathogenic fungi. The research was conducted at Physics Center Krumbate, Aceh Barat Daya District from July to August 2020. Statistical analysis used a completely random design method with 6 levels of treatment and 3 replication. The treatment were carried out by immersing the Barbonimos gonionotus eggs into Calotropis gigantea leaf extract, namely A, without extract, B, 400 ppm extract, C, 800 ppm extract, D, 1200 ppm extract, E, 1600 ppm extract, and F, 2000 ppm extract. A total of 25 eggs that had been infected with the fungus were put into a jar containing 100 healthy eggs and installed an aerator and left to soak for 6 hours until all the eggs were infected. After the eggs were infected, all were immediately transferred to its container that had been given solution of Calotropis gigantea leaf extract according to test concentration of its treatment. Soaking in the solution for 15 minutes and fish larvae that have hatched are observed for survival for 14 days. Now, the result analysis you can see at table 1. Calotropis gigantea leaf extract was effective in suppressing fungal growth on fish eggs. The result of the ANOVA test of research data show the immersion of the leaf extract Calotropis gigantea had a significant effect on egg hatchability and survival of Barbonimos gonionotus larvae infected with the fungus. from the data in table 1, it is known that the hatchability of Barbonimus gonionotus fish egg during the study was 39 until 88%. The hatchability of fish egg in this study was higher than the stat by Agustin and Raharja, namely that the hatchability of fish was generally only able to produce 10,000 eggs with a low hatchability of 22%. In this study, the concentration of Calotropis gigantea leaf extract 800 ppm result in the highest hatchability of fish egg and 88%. This prove the antifungal content contained in the leaf of extract with concentration of 800 ppm is optimal in inhibiting the fungal infection of Sapropelagnia sp on the eggs of fish. Several studies have shown the presence of active chemical compound in the leaf of extract as flavonoid, steroid, tannin, and spannin. Okay, based on the result of the study, it can be concluded that the use of Calotropis gigantea leaf extract significantly affect the hatchability and survival of Barbonimus gonionotus larvae, where the concentration of 800 ppm produce the highest value for hatchability of fish egg 88% and survival rate 92.54%. Okay, thank you, Mr.
Okay, thank you very much, Ms. Iko, for your presentation of your research. For the next presenter is Mr. Dedi Fazrian Shah. Let me share your PPT.
Thank you very much to Ibu Siska Melisa. Assalamualaikum warahmatullahi wabarakatuh. A very good afternoon to everyone. Alhamdulillah, in this opportunity, I'd like to present my poster presentation entitled "The Effect of Dietary Caulerpa lentivera, or sea grape, supplementation on growth performance and survival rate of milkfish channels frog scale." So in this research, in this study, we are going to investigate the use of sea grape flour or Caulerpa lentivera powder on supplemented in milkfish diet in different concentration, like treatment A, control, no supplementation; treatment B, supplementation at 10 gram per kilogram; treatment C, supplementation of sea grape at 20 gram per kilogram; and D, supplementation of sea grape at 30 gram per kilogram; and 5, treatment E, supplementation of sea grape at 40 gram per kilogram. So in this study, we were using the method, the research parameters like absolute line gain, absolute weight gain, specific growth rate, feed efficiency, and survival rate. And then we got result. The best result that show in the treatment C show the significantly better among other treatment, especially in growth performance like absolute length gain, absolute weight gain, specific growth rate, and feed efficiency, but no significant change or no significant effect on survival rate. The second layer, please, Bu Siska. Okay, in this picture, figure 1 and figure 2, we can see that there are the differences of a length gain and weight gain for 49 days of milkfish rearing. So we conclude that the supplementation of sea grape flour, Caulerpa lentivera, significantly affected the growth performance but no significant effect on survival rate of milkfish. So we can recommend that the use of 20 gram per kilogram diet to improve the growth performance of milkfish channels. Thank you very much. See you again.
Okay, thank you,
Assalamualaikum
Mr.
warahmatullahi wabarakatuh.
Yeah, waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Dedi, for your presentation. Okay, we move to the next presenter is Miss Irma. Okay, let me share your poster.
Okay, thank you for moderator for giving me the time for present my poster presentation. My name is Irma Dewianti and my title of the paper is Biodiversity of Megabenthos and Coral Reef Condition in Tuan Island, Aceh Besar. That we know that megabenthos is important role for maintenance of the coral reef ecosystem and also for the food web process. So this is very interesting to learn and to study about megabenthos because in the Tuan Island also have a good condition of the coral reef, so that we know that megabenthos inhabit in the coral reef ecosystem. The objective of this study was to analyze the megabenthos abundance and their composition inhabit at coral reef ecosystem Ujung Pancu subdistrict Aceh Besar, and then to analyze the percentage of live coral cover in Ujung Pancu subdistrict Aceh Besar. For the method implementation, we conducted in July to August 2020 in Pulau Tuan Island, and we can see from this map that we have 4 direction: west, south, east, and north. This is for the sampling location, and also for the conducted the megabenthos, we use with the Bell transect method. This is the very common applied to other megabenthos target. This is some megabenthos target which references to LIPI. There are 8 megabenthos target in the study area. For the result, we found all the megabenthos target, 8 species, and we can see from this table. Yeah, for the Drupella, this is always found in each station. And also from this table 2, we can see that diadema stossum is the highest abundance in study area. For the biological index, we can say that the community structure in the area for megabenthos is medium because the diversity index was medium, the affinity index was medium, and also the dominance index low but to medium, but we can say medium. But overall, we can say the community structure is medium category. For the percentage of coral reef, we can see that in the station 1, 2, no, station 1, this is good condition. From the regressive linear, we can see that the megabenthos abundance, increasing, but the coral reef not increasing. So this is maybe not high correlation between megabenthos abundance and also live coral reef. For the conclusion, there were 8 species of megabenthos target in Tuan Island: Acanthus terpelanci, Lynchia, Diadema stossum, Holoturia, Tridacna, Drupella, Trochus, Panulirus, and then the total 190 individual of megabenthos with the abundance strength from 0.18 to 0.552 individual per meter square. And also, the highest coral cover was station 1, followed by station 3, 4, and 2. And the last, the correlation between the percentage of coral reef cover and megabenthos abundance has a positive correlation, but not too high. We can conclude was low. Thank you, Bu Siska.
Okay, sorry, it's mute. Okay, the next presenter is me. So today I will present my research also. It's about effect of turmeric and formic acid on the growth, feed conversion, and survival of Lithopenaus paname. So in this research, we have the objectives to determine the effect of turmeric and formic acid in feed on the growth, feed conversion, and survival rate. And the methods, there were 4 treatments. A with the turmeric and formic acid, B turmeric 2 gram per kg, and C formic acid 1 gram per kg, and D turmeric 2 gram. The combination turmeric 2 gram and formic acid 1 gram. Okay, for the results we can see the so there are no there are no significantly affect the growth, survival, and feed conversion. We can see in this diagram. But however, there was a tendency for higher survival values for shrimp fat with formic acid supplementation and feed with a combination of turmeric and formic acid. Okay, I think that's all for my presentation. Thank you very much, and we move to the next presenter, Siti Maulida. Okay, Miss Siti, I will share your poster. Okay, wait a minute.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for time that gave to me. Let me introduce myself. My name is Siti Maulida. I'm from Marine and Fisheries Faculty at Aquaculture Department, Syakuala University. In this opportunity, I would like to present about my research by the title Length-Weight Relationship of Penaeus indicus and Penaeus merguensis in the Mangrove Ecosystem Area of Langsa, Aceh, Indonesia. Penaeus indicus and Penaeus merguensis are the most common commercial species found in the mangrove ecosystem of Langsa City. The growth pattern of these two penaeid shrimps are strongly influenced by environmental factors such as temperature, pH, dissolved oxygen, salinity, availability of food, and predators, which ultimately determine the abundance and cage of commercial adult shrimps obtained. The present study aims to investigate the length-weight relationship of P. indicus and P. merguensis in the mangrove ecosystem area of Langsa City, Aceh, Indonesia. The sampling was conducted in November 2018 with the stratified random sampling method. A total of 124 shrimps consisted of 92 samples P. indicus and 32 samples P. merguensis. The results showed the growth pattern of two penaeid shrimps was categorized as negative allometric. The highest B value is shown in P. merguensis with B value of 2.87, while P. indicus 2.55. The value of the relative weight condition factor of the two shrimps indicate a value of greater than 100. It is concluded that the water of the mangrove ecosystem in Langsa City are still able to support the survival of P. indicus and P. merguensis. We would like to thank the Share Resources Joint Solution Program for supporting funds to conduct this study. We are also grateful to Bali Jurong for the cooperation in sampling during the research. Thank you for all your attention. We all know no one perfect. I apologize for my mistake and my broken English. I hope the presentation easy to understand. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Miss Siti Maulida. It's not a broken English. Okay, thank you very much for our presentation. So now, because the time is very limited, now we will move to the closing ceremony and also this oral or poster announcement in that session. So please, our presenter or participant to move to the link in the group. I think we already shared the link in the group. Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you so much. See you soon.
Thank you.
Don't forget to the closing ceremony, please.